Okay, so welcome everyone to the 52nd lecture of Dr. Hyder Step 1. Before we begin today's lecture, can we just make sure that you guys can hear my voice? If you guys can hear my voice, can I get a yes on the chat box, please? Okay, very good. Okay, hope you guys are doing well. Hope you guys had a good day yesterday. Before we begin, can we take into notes of how many questions you guys were able to do successfully from UWorld yesterday? 33. Okay, not bad. Everyone else, how many questions did you guys do successfully? If you guys do not answer, then I would expect you guys did nothing, no questions. That would be really, that would not be really good considering you guys will have your exams in a couple of months. 20, that's very good. Anyone else? Anyone else? 20. Okay, not bad. Out of those 20 questions or 33 questions, how many questions did you guys get right? And were those questions a mixture of all the organ systems? How many correct answers were given? Welcome to all the students who are joining right now. We are just discussing about the amount of questions you guys were able to do successfully yesterday after we were done with the lecture. How many questions did you guys do yesterday? %80 correct in random. That is very, very good to know. 80% correct in random is very close to doing very well. Okay, and anyone else? What are your scores and percentages in the U world, please? About 29. If you get 29 correct out of 33, that is exceptionally good. That is very, very good to know. Thank you so much, Dr. Iman, Dr. Hossam, for letting us know your scores. Anyone else? Dr. Muhammad, Dr. Gianni, how many questions are you guys getting right? And to the rest of the students, how many questions are you guys doing? %75 from Dr. Harry. Very good. Okay. Okay, very well. Okay, so now that we are done with the organ systems, from today, what we are going to do is we're going to jump into the basic science portion, and the first chapter is genetics and biochemistry, and that's what we're going to do. We're going to start with that one. Um, is everyone ready to jump in without wasting any more time? Because it's quite a long topic, and I want to make sure that we start early. Okay. No problem. I just wanted to know how many questions you got right out of the 20 questions which you did, Dr. Mohammed. Okay, no problem. Okay. All right. Okay. So what I'm going to do is, without wasting any more time, I'm going to start the lecture on genetics and biochemistry. Before we begin genetics and biochemistry, first and foremost, how complicated and intimidating is genetics and biochemistry? Anyone? Very, really hard. Okay. What if I were to tell you that genetics and biochemistry is not actually hard at all? It's all about practice. What do I mean by that? The fact that genetics and biochemistry, it's not hard, it's about practice. This means that if after doing first aid with us right now, if you don't do the UWorld questions from genetics and biochemistry, then you will forget the knowledge very, very fast, very fast. Okay. So every day after we are done with the discussion on genetics and biochemistry, if possible, at least try to do 5 to 10 questions from biochemistry and genetics. That is the practice which I'm talking about. And when you get the hang of it, you will realize that each and every question from genetics and biochemistry, they keep on coming back and forth with the same pattern. So there's a pattern which you need to get hold of, and then you will realize that it's not actually very, very difficult. So, and with that being said, there's another thing that is genetics and biochemistry. It is sort of difficult in the sense that we will not be able to get into the absolute depth of genetics and biochemistry, meaning that if you try to understand each and everything, that is very good, especially from first aid. But there will come a time when we might ask ourselves that exactly why is this happening or why is that happening, and then we are going to go into deeper genetics and deep biochemistry, and what that will do is that will deviate away from our from our knowledge which are needed to do well in the scope of doing good in USMLE step 1. So we are going to study exactly what's needed, and exactly we are going to tell you how you can break those knowledges down into easy digestible form so that it's easy for you to review, recap, and understand everything. Okay. So with that being said, are we all ready to begin? We have 25 students as of right now. It's 9:11 a.m. Okay, there's not really much to revise and recapitulate because we're done with the organ system. So the first segment of our lecture today is not going to happen. From tomorrow, we're going to do the revision and recapitulation of today's lecture. Okay, so right now we are going to begin. Okay, let's start. Let me share my screen. Okay, okay, all right. Okay, so we are going to begin with biochemistry and the molecular portion of biochemistry, that is genetics. That is exactly what we're going to begin with. Really excited over here. Nothing to be worried about. Easy stuff. I'm just going to tell you exactly what's going on over here, how you are supposed to phase these questions in the step 1, and in no time we will be done with this very soon. Having said that, we will come across certain pathways. Okay, we will come across certain pathways, like we will come across de novo, pyrimidine and purine pathways, and then purine salvage pathways. So for these pathways, what I need you to do is, when I would be drawing these pathways on a blank page, I need you to have a notebook or a piece of paper ready on the side. So when I am drawing something, I need you to draw the same thing. Okay, so what that would do is, if you draw the same thing which I am drawing on the blank page, at the end of the day, you can keep those notes. Okay, you can keep those notes, and either you can post it on your wall or you can keep it on your notebook, and that would allow you to keep all the pathways in one book. So if you have to do a quick revision and recapitulation, most of the questions are usually from the pathways. Most of the questions are usually from the pathways of the genetics and biochemistry. Very handful amount of questions are expected from the text. We're going to tell you what are those questions. But when we are doing the pathways, please make sure that you make your own notes for the pathways. Keep a notebook. Okay, when we are writing something, write it down so that when you want to do a quick revision and recapitulation, you can go through the pathway book which you have created. Okay, have I been clear? Have everybody understood what I just said? Okay, good, very good. Okay, so without further ado, let's begin today's lecture. First of all, the first thing that we're going to study today is the structure of chromatin. And before we jump into this, let's do a small introduction to genetics, and that's that. Genetics is basically the study of your genome and all the structures that are in it, right? So if this is basically a cell, right, and then this is the nucleus, then the nucleus is nucleolus, and then over there in the nucleolus we have what we say is the genetic structure. We have the DNA, all right? We have the DNA. right? So DNA is basically deoxyribonucleic acid. It's a nucleic acid over there in the DNA. In the DNA, okay, in the DNA, what you have is you have small pieces of informations. You have very small pieces of informations, right? You have small pieces of informations in um, in the form of in the form of what we say is chromosomes or chromatins, right? And those chromosomes, they contain, they are filled with even minor informations. Okay, they are filled with even minor informations that are known as genes, right? That are known as genes. And in those genes, okay, and in those genes, you have each and every information at each and every position. So first of all, you have the DNA, and the DNA, okay, you have the chromosomes, right? Chromosomes have DNA. In those DNAs, you have genes, right? And those genes are located, these genes, each and every gene can be categorized into alleles. Those alleles are located at a single position of the gene, right? And those positions are known as locus. These are some minor terms which we have to get accustomed with. Have we understood this? Have we understood this small piece of information before we even begin? We have chromosomes, right? How many pairs of chromosomes do we have? Chromosomes. How many pairs of chromosomes do we have? We have 23 pairs of chromosomes. That's correct. Each and every chromosome, it contains the DNA structures. Those DNAs, they have small pieces of information that are called genes. Those genes are located. Each and every gene is known as an allele, which is located at a different position. The positions are known as the locus. That's that. So right now we're going to study of how a chromatid or a single chromosome is formed from the gene. Uh, from the gene, meaning that how the DNA will structure itself to form a chromosome. That's exactly what we are going to study, how that is happening. And the reason why this is important is because clinically, we can use the activation or deactivation of chromosomes for different purposes. Okay, so clinically, it's important. That's why we have to study this. First of all, the structure of the chromatin. Chromatin is basically DNA. It exists in the condensed chromatin form to fit into the nucleus. So DNA is basically condensed because DNA is such a long piece of information. If it was not condensed into a smaller piece, it would not be able to fit inside the nucleus. So the DNA is condensed to form a small chromatin form to fit inside the nucleus. The DNA, as you can see over here, it loops twice around the histone. Histone is basically, it's nothing but a protein. Okay, so histone, it it loops twice. So the DNA is condensing. After it condenses, the DNA, it loops twice around the histone. And after it loops around the histone, it forms what we call as a nucleosome. Nucleosome are basically these structures over here. These structure meaning that this beads on a string appearance, these beads on a string appearance with each and every histone containing two loops of the DNA. This is known as a nucleosome. Okay, then there are different types of histones. You have H1, H2A, H2B, H3, H4, right? So each and every one is a histone over here. H1, it binds to the nucleosome and to a linker DNA and thereby it stabilizes the chromatin fiber. So basically, the H1 will bind to the nucleosome and it will bind to the linker DNA. It will bind to one nucleosome and then it will bind to the another portion of the DNA, and then it will stabilize the chromatin fiber over here. Till now, from this paragraph, you have no questions which are coming. Okay, no questions will be asked from this paragraph, and this is just for your understanding, that's all. Okay, next one. Next one is The DNA structure has a negative charge and a positive charge. Okay, the DNA has a negative charge and a positive charge. The phosphate group. Okay, so what I need you to do is I need you to write this over here. DNA. Okay, DNA. Negative charge is from the phosphate. Okay, negative charge is from the phosphate group. This is an NBME question. This is a question from the NBME. Okay, the phosphate group, it gives the DNA a negative charge. Lysine and arginine gives the histone a positive, give histones a positive charge. So you have the DNA, which is the negative, which by the help of the ion, that is phosphate, and histones, they have a positive charge. So write down over here that histones, they have a positive charge. Positive charge. Why? Lysine and arginine. Okay, lysine and arginine. So write this down in your book. These are NBME questions. Okay, questions from the NBME. That's that. Okay, so lysine and arginine are basically, they are amino acids. Okay, they have a positive charge, and the DNA, they're given a negative charge by the phosphate, and that's that. In mitosis, the DNA will condense to form a chromosome, and the DNA in histone synthesis, it occurs during the S phase. This is also a question. This is a question. At which phase of the DNA cycle does DNA, at which phase of the mitotic cycle does the DNA and histone synthesis occurs? And it's at the S phase. And we have talked about this before, that in the S phase, you have the DNA which condenses, and then you have the structures which are getting ready to separate, right? So at the S phase, the DNA and the histone the DNA and histone synthesis will occur at the S phase. Okay, next one. Next one is the mitochondria. They have their own DNA, which is circular, and it does not utilize the histone. The fact that mitochondria has a circular DNA, give this 5 stars. Okay. Next time you receive a question on NBME that which organelle has a circular DNA, your answer is mitochondria. Circular DNA is mitochondria. So mitochondria has a circular DNA and it does not utilize histones, meaning that the DNA in the mitochondria, it does not need the histones to compact the structure into a chromatin form. It can just compact by itself because the DNA is a circular DNA. Okay. So far, have we understood all the questions which will be received from this paragraph? Have we understood all the questions that will be received from this paragraph? Okay. Okay. Okay, so DNA, the positive charge of the histones, which amino acids are responsible for the positive charge of the histone? Which amino acids are responsible for the positive charge? Yes, very good. It's lysine and arginine. Which ion is responsible for the negative charge of the DNA? Okay, good, very good. Then DNA synthesis occurs at which phase? DNA synthesis and condensing. Good. Which organelle has a circular DNA and does not contain any histone? How many loops does the DNA make around a histone to form a nucleosome? Okay. Okay, good, very good. Okay, all right. So we are going to discuss this diagram after the end of this page because we will study these things first and then we will understand what they're talking about. After that, we will jump into this diagram. Next one is heterochromatin. What is heterochromatin and euchromatin? These are two terms which we have to understand. Okay, heterochromatin is basically the condensed DNA. DNA, when this is very condensed, it appears darker on the electron microscope. As you can see that over here, it is labeled H. This H is the condensed DNA. This extremely condensed DNA is known as heterochromatin, and it's sterically inaccessible, meaning that this heterochromatin is basically inactive, meaning that if you try to if the genes in the DNA they decide to form a protein, okay, for example, go through the process of transcription and translation and then form a protein structure, this will not happen because heterochromatin are inaccessible. So this is also another piece of information, okay, that is transcriptionally inactive. Transcriptionally inactive is heterochromatin. Heterochromatin is transcriptionally inactive, and transcriptionally active is euchromatin. Transcriptionally active is euchromatin. Okay, so basically this is transcriptionally inactive, meaning that for a DNA to be transcriptionally inactive, one thing has to go up, another thing has to go down. Always remember this. When a single portion or when a certain portion of the DNA is methylated, when something is methylated in the DNA, it is inactive. When something is acetylated, it is active. So if heterochromatin is inactive, then there should obviously be increased methylation and decreased acetylation. Okay, decreased acetylation. The easiest way you can remember this is M for mute and A for active. A for active. So next time, if you receive a question in NBME where they are talking about acetylation, try to understand that they are directing you to a question where they are talking about DNA activation. If they are talking about methylation, then they are directing you to a question where they are telling you that a single portion or the DNA is inactive. It's not going to work. It will not take place in the process of protein synthesis. That's exactly what they are trying to tell you. So M for mute or methylation, A for active or acetylation. Okay. Next one. Next one is heterochromatin is highly condensed, so HC, and the bar bodies may be visible on the periphery of the nucleus. Not high yield. Okay, these are not high yields. I'm pretty sure you guys know exactly what bar bodies are. Bar bodies are inactive X chromosomes. That's all. Okay, that's that. Having said that, let's move on to euchromatin. Euchromatin is less condensed, and it appears lighter on electron microscope. And the euchromatin is this portion over here. Is this portion over here? This lighter portion. This lighter portion is known as euchromatin, and it's transcriptionally active. If it has to be transcriptionally active, what would happen to methylation and acetylation? Can I get some fast answers, please? If euchromatin is transcriptionally active, what would happen to methylation? So increased in acetylation and decreased in methylation. Very good. Okay, very good. And euchromatin is expressed, meaning that euchromatin is the chromatin which can undergo DNA activation and the process of protein synthesis. That's that. So that's all. Okay. So these are the informations over here. The reason why I'm asking you to write these down over here is that when you go through this page again, you know exactly what to look out for, so that it will be easy for you to revise this page as fast as possible. Okay. Do you guys see a lot of students on Usmle Step 1 preparation forum who keeps on telling everyone that who wants to revise first aid with me in 1 week, or who wants to revise? Is there anyone who is there who wants to revise? Have you ever wondered how that's possible? How they do this? That's because they have pre-read the first aid, and they write down small pieces of information that exactly which they go through, right? And that's exactly what we are doing right now. So we are writing the small pieces of information over here on the pages, so that next time when we go through them, it will be faster for us to finish the first aid. Okay, that's that. Next one. Next one is DNA methylation. DNA methylation, it changes the expression of a DNA segment without changing the sequence. So basically, DNA methylation is you add a methyl group. The methylation is it's usually within the gene promoters. Gene promoters are basically known as CPG islands. CPG islands, the word CPG, are promoter regions within the gene. So the genes, they have a promoter, meaning that if something goes and binds to that portion of the gene, then genetic promotion will happen, meaning that there will be the process of protein synthesis can begin, very simply put. So over there, if on the promoter region, if you go and attach a methyl group, then it will stop genetic transcription, meaning that it will stop protein synthesis. The fact that the methyl group is added to the gene promoter CPG island, this is high yield for NBME. Okay, NBME. So meaning that basically, if you want to stop DNA, if you want to stop protein synthesis, and you are a research scientist, right? And let's say your research director asks you at which portion of the gene would you add the methyl group in order to stop the gene transcription. Your answer should be the gene promoter sequence. That is known as CPG islands. CPG islands. Okay, so that's high yield. Next one is the DNA methylation. These are the changes of the DNA segment without changing the sequence, meaning that you are not changing the sequence of the DNA. You are just stopping the DNA from making a protein. That's that. It's involved with aging, meaning that with age, what happens is that your DNA, it stops um, producing new protein, right? And that's why we age and we stop with regeneration. So that is because our DNAs are constantly being methylated. So if we found a process to stop this methylation of the DNA, would there be a possibility that we could stop the aging process or slow down the aging process? Yes or no? Yes or no? Did you guys understand what I just said? With aging, what happens is, with aging, DNA methylation occurs. So with DNA methylation, what happens is with DNA methylation, the DNA, they stop transcribing new protein, right? So they stop transcribing new protein, and then what happens is you age and you stop regenerating. So if anyone found a way to stop this DNA methylation within the genetic sequences and the CPG islands, then we could actually slow down aging. That's that. It's involved with aging. It's involved with carcinogenesis, right? With formation of carcinomas. DNA methylation is also involved. It's involved with genomic imprinting. This is a process we will talk about in a second. Transposable element repression. Repression of transposable elements. Okay, we will also talk about transposable elements in a second, not right now, and also in the inactivation of X chromosome. So basically, they're talking about all the different types of processes where DNA methylation is involved. Okay, it's not very difficult for us to realize that in processes such as aging and carcinoma formation, you have DNA which does not form any new protein anymore. It stays what it is. So that's that. Important thing to take from this paragraph over here is CPG islands. That is the gene promoter sequence from where the methyl groups will go and bind. And when they bind to the CPG islands, they will have stoppage of DNA uh, of protein synthesis from the DNA. Okay. Next one is histone methylation. Histone methylation, it usually causes reversible transcriptional suppression. So right now, instead of the methyl group being bound to the DNA, the methyl group can go and bind to the histone. And when it goes and it binds to the histones, they get what we call is reversible transcriptional suppression. Reversible transcriptional suppression, meaning that the transcription and translations which are responsible for protein synthesis, that transcription can be stopped and it can be reversible, meaning that this stoppage of transcription, it can be reversible to initiation of transcription once the methyl group is removed from the histone. So reversible transcriptional suppression. Okay, this is a high yield question in NBME. Once again, most of the questions are from the genetics are usually from the NBMEs because they try to dig it deep, whether you have taken into the knowledge which you have read in first aid. So reversible transcriptional suppression. Reversible transcriptional suppression. Which molecule is involved and which group is involved in this process? The molecule that is involved is histone, and the group that is involved is methyl group. Methyl group. Okay, methyl group. That's that. Next one. Next one is histone. Next one is histone acetylation. Okay, should we know these processes? What processes are we talking about? What processes are we talking about over here? Dr. Allah? No, no, you should not know these processes. Okay, these are not high yield. These are beyond the scope of step one. Okay, okay, all right. So reversible transcriptional suppression that occurs because of histone methylation. Okay, next one is histone acetylation. Histone acetylation, basically acetylation is A for active, A for acetylation. Histone acetylation will make the DNA active. And how do we acetylate? Basically, histone acetylation is the removal of histone's positive charge. Histone's positive charge. What were the amino acids that was required for giving the histone the positive charge? The positive charge of the histone? Do you guys remember the name? It was lysine and arginine. Right now, if we remove lysine and arginine, if we remove the lysine and arginine, that will remove the histone's positive charge. And what would happen is histone, if we remove the positive charge, the DNA can get relaxed, right? Because the DNA was condensing, and the more condensed, the less active. As we just studied, that heterochromatin, they're more condensed, that's why they're less active. So we have to make the DNA less condensed so that they can uncoil, right? I mean, so that they can coil and they are available for genetic transcription. So when we remove the histone's positive charge, then DNA can relax, coiling can occur, and transcription can increase. So basically, histone acetylation is DNA relaxation and removal of the histone's positive charge so that transcription can increase. The exactly opposite process is histone deacetylation. Deacetylation is removal of the acetyl group. What happens when we remove the acetyl group is that the DNA, it tightens. Once again, it tightens. And when the DNA tightens, it supercoils and it coils back into the condensed heterochromatin form. And then the transcription is decreased, as simple as that. Okay. Okay, having said that, can I give you guys exactly 2 minutes to learn all this information over here before we move on to the next page? That is this one. Because we want to make sure that all the information from first aid are learned right away so that the rest of the day can be spent on doing questions. Okay. And since this is high yield information, and we have written down exactly all the things you need to know from this page over here. Before we jump into the revision, next time when you guys go through this page, will this be easy for you to go through very fast because now you know exactly what you need to study? Yes or no? Yes. Okay, so this is how we are going to proceed with genetics. Are we clear? This is exactly how we are going to proceed with genetics so that next time when you come across a page, you know exactly what you need to go through. Okay, so that's that. So having said that, it's 9:39. I'll give you guys 2 minutes. I'll give you guys exactly 2 minutes. Please learn this information and I'll ask you. And once I've understood that you guys have you guys can produce the knowledge, then we can move on to the next page, this one over here. Okay, so 2 minutes on the clock. Let's do this. Okay, is everyone ready? My first question is, my first question is, um, DNA, which protein does DNA use to loop around to form a condensed structure so that it can fit inside the nucleus? Fast answers, please. Histone, very good. Okay, histone. Now, which ion is responsible for giving DNA the negative charge? Fast answers. Phosphate, very good. Which amino acids are responsible for giving histones a positive charge? Lysine and arginine, very good. At which phase of the DNA does, at which phase of the mitotic cycle does the DNA and histone synthesis occurs? S phase, very good. Which organelle has a circular DNA and does not use a histone to form a condensed structure? Very good, mitochondria. Which type of chromatin is stereotactically inaccessible and inactive? Heterochromatin or euchromatin? Very good. Euchromatin, if it has to be active, then is it highly methylated? Yes or no? No. Is it highly acetylated? Yes, yes or no? Yes. Okay. Histone methylation, DNA methylation. Can you guys tell me the name at which region does histone, or DNA methylation occurs? CPG island promoters. Very good. Next one. Next one is histone methylation. Is it if we methylate the histone, does that mean that the transcription is stopped indefinitely? No, it's reversible transcriptional suppression. If we add a histone to, if we acetylate a histone, are we removing the negative charge or are we removing the positive charge? The positive charge of the histone. Okay. What happens to the DNA coiling when we add, or what happens to the DNA coiling when we remove an acetyl group? When we remove an acetyl group, tightens. Very good. Tightening. Okay, good. And there's one last thing which you guys have to realize from this one over here. Okay, so very good. You guys could answer most of these questions correctly. Okay, next one. The thing that you have to understand, and there's another piece of question which is hiding in this diagram over here, is that the nucleosome, it consists of H2A, 2B, H3, and H4 histone. Okay, this is high yield. So out of all the histones, H234 is responsible for the nucleosome. So this is another question from the NBME. That's that. Okay. Okay. And H1 is the linker. H1 is the linker molecule. So basically the nucleosomes, they contain H234, 2A, 2B, 3 and 4, and H1 is the linker over here. Okay. So do not forget the functions of these histones. Okay, next one. Next one that we're going to go to is this page over here. There are multiple questions from this page, and very easy to understand, very high yield nonetheless. Nucleotides. What is a nucleotide? Before we understand what a nucleotide is, we have to understand what a nucleoside is. Nucleoside is basically a DNA base plus S for sugar, meaning deoxyribose. So a base with a deoxyribose. So deoxyribose is the sugar portion over here. So if we try to see the structure of a nucleoside, okay, the structure of a nucleoside, this one over here, okay, so you have this structure, that is this one, that this is the base, okay, that this is the basic structure, and this, then you have the deoxyribose sugar portion. You have the deoxyribose sugar. Deoxy meaning two hydroxyl groups attached. So deoxyribose sugar over here. Okay. Are we clear about the nucleoside structure? Not high yield. This is just for your understanding. Okay. So base plus sugar is nucleoside. That's what I wanted to come across. That base plus sugar is nucleoside. Next one is base plus sugar plus phosphate is nucleotide. So T for phosphate, S for sugar. When you understand what a nucleoside is and a nucleotide is, then next time when you come across these questions, then you can identify that nucleoside does not have 3', 5' phosphodiesterase bonds. Nucleotides, on the other hand, they have a 3' to 5' phosphodiesterase bond. And these bonds are really important in the fact that they are responsible for if you want to get rid or if you want to break down the DNAs, then you have to go and target these bonds over here. So these are actually stronger portions. So nucleotides are the stronger portions. That's that. Next one. Next one is the nucleotides. Basically, there are purines, and then there are pyrimidines. Okay. First of all, let me ask you guys: do we have any student over here who's a big fan of gold, especially the female students? Big fan of gold. Okay. Do we have any students over here who loves eating the pie? Who loves pies? Pie. Okay. No one loves pie. Does anyone love gold? Okay. Is gold pure better or unpure gold better? Pure gold is better or unpure gold is pure gold is better. So basically, if I tell you that pure, the reason why I am also writing this, first aid also has this mnemonic, but I'm also doing this because this will help you. Okay, pure as gold. Pure as gold. Basically, if you are ever confused that whether the nucleosides, that is adenine and guanine, are they purines or pyrimidines, then this mnemonic, pure as gold, will help you identify that adenine and guanine, these are purines. These, both of them are purines. And next one is, I asked whether you guys love having the pies. Some of you guys said yes, some of you guys said no, but nonetheless, if you have to have the pie, you have to cut the pie. Cut, C-U-T. Cut the pie. Okay, cut the pie. Basically, basically meaning that C for cytosine, U for uracil, and T for thymine. These threes, they are the pyrimidines or the pyrimidines. Okay, so there comes a lot of time when you are giving the exam for so long, a basic question that whether this structure is purine or pyrimidine, that can get you confused. But if you remember that golds are pure and you have to cut the pie, then you will never forget this. So that's the reason. So pure as gold and cut the pie. And this is also a high yield question. It's a very easy question, but it can be pretty difficult if you're not 100% sure. That's that. An NBN question over here. Okay, next one. Next one is the purines. They have 2 rings, and the pyrimidines, they have only 1 ring. Okay. By that we mean the base structure. Okay, so the purines, they have 2. The pyrimidines, they have 1. Okay, next one. Next one is if you remove an amino group from the cytosine, the structure that they will form is uracil. If you remove an amino group from adenine, the structure that they will form is hypoxanthine. These are all basically structures which are responsible for the metabolism. That's that. Next one is if you remove a structure from the guanine, the structure that they will form is xanthine. And if you remove the structure from 5-methylcytosine, the structure that they will form is thymine. So let's use a mnemonic to remember this piece of information. Okay, let's come over here. So what we're going to do is, okay, next one. So what we're going to do is, okay, is you cycle, then am hypo, then Good Christmas, and okay, me taco. Okay, you cycle for the fact that cytosine, it is converted to uracil. Am hypo for adenine to hypoxanthine. Good Christmas for guanine to xanthine, and metaco for methylcytosine to thymine. Can I give you guys 1 minute? Okay, I'll give you guys 1 minute because these deamination reactions are important, especially for NBME, especially for NBME. You will not receive these questions in UWorld. Okay, you will not receive these questions in UWorld. So for NBME, this is important. Okay. So these are a little bit of these are a little bit um, I mean, these mnemonics are a little bit out of the ordinary, meaning that they're a bit random. Okay, but to be honest, they have helped me out in the past to remember the deamination reactions. If you can come up with a better mnemonic, that's up to you. But if not, then you can use our mnemonic. But no matter what you do, please try to remember the deamination reactions, meaning that if you remove the amino group from cytosine, structure you're going to end up with is uracil. If you remove a structure from the adenine, the structure you will end up with is hypoxanthine. Same thing, from guanine comes xanthine, and from methylcytosine comes thymine. That's that. Okay. NBMA is more close to the real exam. I always believe that UWorld self-assessment 2 is the closest to the real exam. NBMEs are extremely difficult. To be honest, they're more difficult than the real exam. We think that the UWorld is the closest to the real exam as possible, especially the score you will receive in UWorld self-assessment 2 is very, very close to the score you will receive in your actual exam. So we ask our students to take the UWorld self-assessment 2 at the final weeks of their exam. Okay. So such things won't come in exam. They might come in the exam. You never know, since they are there in the NBME. Okay. Questions in NBME, they're made by the examiners, or they are added in the NBMEs from past questions. So what we know is that this is high yield because there are reasons why you need to understand this, because in the future you will have pharma which will be linked up to this knowledge. Okay. NBME and Kaplan is taken from the real exam. Yes, they are, most of them, but to be honest, NBMEs are made exceptionally difficult because they add all the hard questions in one exam together. So that's that. The real exam is not actually as difficult as the NBME. Okay. Are we done with this? The deamination reaction? It's been more than 1 minute. What happens when you remove an amino group from cytosine? You cycle. Uracil, very good. What happens when you remove an amino group from adenine? Am hypo. Very good. What happens when you remove an amino group from guanine? Good Christmas. Very good. What happens when you remove an amino group from methylcytosine? Metaco. Very good. Timing. Okay. So it will be a little bit difficult to remember this because the mnemonics are a bit random, but hopefully you guys do, because you guys will need this. Okay. Next. So this is a piece of information that is high yield from over here. Then the next one is Then the next one is the 5' end of incoming nucleotide, they bear the triphosphate, which is the energy source of the bond. We will talk about the 5' and the 3' in the DNA replication. So that's that. But when we do, we will talk about the triphosphate group and the triphosphate bond. And the phosphate bond, the triphosphate bond is a very strong bond between the DNAs. That's that. Okay, next one. Next one is, this is a very, very important piece of information over here. That is, out of all the bonds, CG bonds, meaning that the bonds that is formed between cytosine and guanine, they are stronger than the AT bonds. That is the bond that is formed between adenine and thymine. You know, like how we keep on saying AT, C, G. And what happens is, basically, if you have a DNA structure like this, right? You have a DNA structure like this. Okay, what's happening over here is that you have A, T, right? And then you have C, G. And this keeps on continuing throughout the whole cycle. A, T, C, G, A, T, C, G. And that's exactly what's going over here, that you have these bonds. Out of these bonds, the bonds that is formed between C and G, meaning that the bonds that is formed between cytosine and guanine, they are way stronger than the bond that is formed between adenine and thymine, because there's 3 H bonds for CG and 2 H bonds for AT. And this is very, very high yield. So what happens is, if you want to break down the bonds of the DNA, you have to target the CG bonds. An increase in CG content, meaning that the amount of cytosine and guanine there are, the stronger the bond is, and the higher the melting temperature of the DNA. So CG is basically like crazy glue. Okay, this is a very helpful piece of information over here, that crazy glue. Crazy glue. This is very, very high yield. 100% you guys will receive this question in NBME, no doubt. Okay, so that's that. This is also another piece of information. Please, if needed, write down CG bonds. CG bonds are more stronger. Okay, CG bonds are stronger than AT bonds. Amino acids which are necessary for purine synthesis. If you want to synthesize a purine, the amino acids that are necessary is glycine, aspartate, and glutamine. And we will talk about this in 1 second when we jump into the de novo pyrimidine and purine synthesis. And over there, we will see that the amino acids which we are using for the synthesis of purines and pyrimidines are these 3: glycine, aspartate, and glutamine. So basically GAG. This is also high yield. Okay, this is also high yield. So once again, 1 minute on the clock, it's 9:59. Okay, please learn all these pieces of information over here. Once we have figured everything out, we will move on to the next page. Okay, 1 minute on the clock. Do you guys realize exactly what you need to study from this page? Are we all clear about this page? The reason why I'm asking is because I will make sure each and every page is mastered. Okay, next time we come across this page, how long will it take for you guys to review this? 1 minute, 2 minutes, right? Am I correct? Okay, very good. Okay, so you have exactly 1 minute. Please review this information over here so that we can move on to the next page. Okay. Okay. What is the difference between a nucleoside and a nucleotide? Fast answers, please. Nucleoside and the nucleotide. Okay. Which types of nucleosides are purines and which are the pyrimidines? Pure as gold, and the rest are pyrimidines. Very good. Which bonds are stronger, AT bonds or CG bonds? Which amino acids are responsible for the formation of purine synthesis? For purine synthesis, glycine, aspartate, and glutamate. Very good. When you remove an amino group from the adenine, what structure does it provide? Hypoxanthine. When you remove an amino group from guanine, what structure does it provide? Very. When you remove an amino group from 5-methyl-cytosine, what structure does it provide? Thymine. Okay, good, very good. And okay, okay. I guess that's that. No other questions. Okay, all right. Let's move on to the next page over here. That is de novo pyrimidine and purine synthesis. Okay, right now, I'm not sure if the students who have joined later have heard what I planned for for the um, for these tables over here. Is that what I need you guys to do is I need you guys to have a blank page on the side or a notebook on the side. So when I write a structure, I need you guys to write the structure so that when we are writing it, you are writing it, and already you guys will know exactly, because when you write something, it's almost as if you have read it 5 to 10 times, right? So and what this will do is you guys will have made by the end of the lecture, you guys will have a notebook which will only contains all these tables over here, right? So this will allow you to review the biochemistry very fast. So that's what I want you guys to do. So without further ado, let's jump into the de novo pyrimidine and purine synthesis. Okay, it's a bit difficult, or that's how it looks like, but after the end of this lecture, hopefully this gets very easy, and you will receive a lot of questions from de novo purine and pyrimidine synthesis. What do we mean by the word de novo? De novo, the word means that you are producing the purines and pyrimidines within the within by yourself, by using the structures that are already present in your system. That's the word. That is the word for which, that is the concept for which the word de novo is used. So first of all, we will talk about purines and pyrimidine synthesis. We know that the purines are adenine and guanine, and cytosine, uracil, and thymine. These are the pyrimidines. So that's that. Okay. So basically, the first structure that is required for the formation. Okay. First of all, before I begin, is everyone ready with their notebook and their pen and paper so that we can begin? Is everyone ready? Okay. Okay, good, very good. Okay, so let's take this step by step. Okay, let's take this step by step. The first structure that is responsible for the formation of this purines and pyrimidine is ribose sugar. So the sugar is ribose 5 phosphate. The sugar is ribose 5 phosphate. Ribose 5 phosphate, this sugar that is already present in your system, this gets converted into a very high yield structure that is known as phosphoribosal pyrophosphate, or PRPP. Phosphoribosal pyrophosphate. And the enzyme that is responsible for converting this process, that is from ribose 5 phosphate to phosphoribosal pyrophosphate, the enzyme that is responsible is known as phosphoribosal pyrophosphate synthetase. Synthetase. Okay. And it's basically a very passive reaction, meaning that no energy is being produced over here. So no ATP or those things are going to happen over here. Okay, so that's that. And no energy is released. This is just a conversion. So ribose 5 phosphate is converted to phosphoribosal pyrophosphate by the enzyme PRPP synthetase. Okay, so right now this PRPP synthetase is a very high yield substrate for completing the purines and the pyrimidine synthesis. So let's talk about the purines first. Okay, let's talk about the purines first, the de novo purine synthesis. So we will talk about the purines at this side. Okay, the purines over here, that is adenine and guanine, right? So we'll talk about the purine. Okay, and what happens is that the PRPP over here, this is converted to, the next steps are very, very easy. PRPP, the next step from PRPP is ionosine monophosphate, IMP, IMP or ionosine monophosphate. And from this ionosine monophosphate, what you have is you have your adenine and guanine in the form of AMP or adenosine monophosphate and GMP or guanosine monophosphate. And this is your purine pathway. We're done 1 out of 2. Easy to understand, yes or no? Ribose 5-phosphate is converted to PRPP by PRPP synthetase. PRPP is converted to IMP. IMP is converted to AMP and GMP. Very easy, right? Okay, good. So we are done with the purines one. We're done with purine. Let's talk about pyrimidine. A slightly, uh, a slight bit more complex than purines, but equally easier. Okay, very easy to be honest. Pyrimidines. Pyrimidines, okay, the pyrimidines over here, the first step of pyrimidine is basically you have the amino acid. Okay, you have the amino acid, that is glutamine. What happens is, this is very easy once you get the processes right. Basically, glutamine is converted with the help of carbon dioxide. Carbon dioxide, that is this is basically released from the metabolic pathways when it will come and bind to the glutamine. This will convert in the presence of a very high yield enzyme. Okay, that this enzyme is known as carbamyl phosphate synthetase. And the last word I'm going to use is 2, and I'm going to use a different one, that is carbamyl phosphate synthetase 2. Okay, focus on the 2. Don't forget the 2 over here. Okay, it's not 1, it's 2. Carbamyl phosphate synthetase 2. And glutamine and carbon dioxide in the presence of carbamyl phosphate synthetase 2 will form carbamyl phosphate. Very easy to understand. So glutamine will bind with carbon dioxide to form carbamyl phosphate. Okay, carbamyl phosphate. This carbamyl phosphate that is formed, carbamyl phosphate that is this form, this is not basically a conversion. This is a reaction. Meaning, what happens over here is that over here you require ATP. Over here, ATP is required, meaning that it's not a passive reaction. You need the active supply of energy to make this conversion to occur properly. So the energy that is required over here is you require 2 ATPs. You require 2 ATP. This ATP is broken down to ADP, and obviously a phosphate group is released, right? Or else how will you realize the energy is gone? So 2 ATP plus phosphate plus this glutamine is also released in the form of glutamate. Glutamate. Okay, glutamate. Okay. Now, so this is the first complex reaction. Over here, if you can do this reaction, the rest of the ones are very, very easy. Then the next one is, do you guys remember we talked about glycine, aspartate, and glutamate? This is the first part where glutamate was used. Okay, glutamine was used. And now the carbamyl phosphate, the next one is the carbamyl phosphate. This is converted to another high yield structure, that is orotic acid or orotic acid. Orotic acid, the fact that carbamyl phosphate is converted to orotic acid, this is done in the presence of another amino acid. Out of those 3, that is glycine, aspartate, and glutamate, The amino acid that is used for this one is aspartate. Aspartate. Okay. So an acid is used to form another acid. Amino acid binds with carbonyl phosphate to form orotic acid. Okay. What happens is this orotic acid, once you have this formation, once this has occurred, then the next thing that happens is basically this PRPP over here, this will come and this will bind with the orotic acid to form a monophosphate like this one. The only difference is the monophosphate over here is called UMP. UMP. And UMP is basically this is formed in the presence of UMP synthase. UMP synthase. Okay. Now, UMP synthase, this monophosphate is converted to diphosphate. So UMP gets converted to UDP and UDP. Okay. And what happens is this UDP once again goes through another reaction over here. Okay. This reaction is a very high yield reaction, and we have talked about this reaction in the past when we studied oncology. That is UDP is converted to DUDP. DUDP. Does anyone know what the small d is called? Why did we add the small d? Anyone? Anyone? Okay. Oh, you guys already said deoxy, right? So that's what it is. So basically, DUDP is a pyrimidine 2' deoxyribonucleotide 5' diphosphate, meaning that it's a deoxy uridine, deoxy. So that's that. So DUDP. Okay. So from DUDP, so from UMP you have UDP, from UDP you have DUDP, and the next structure that forms from DUDP is DUMP once again, right? And from DUMP, what you have is you have DTMP. DTMP. Now, the process of DUMP being converted to DTMP, okay, DTMP, this occurs in the presence of a reaction, that is tetrahydrofolate is converted to dihydrofolate, and dihydrofolate gets converted back to tetrahydrofolate, and tetrahydrofolate gets converted to methylene tetrahydrofolate. Methyl tetrahydrofolate. All you have to remember that there is a folate reaction going over here. That's all you have to remember. And whenever there's a folate reaction going over here, the enzyme that is required is dihydrofolate reductase. Dihydrofolate reductase. And this conversion is responsible for converting DUMP to DTMP. And also, there's also another enzyme over here, which we also discussed previously in the oncology drugs, that is thymidylate synthetase. Thymidylate synthetase. Okay. So you already have the T, you already have the A, you already have the G. What you have is you have the missing C. The C is missing. That is the cytosine. So basically, and fortunately, the cytosine is directly produced from the UDP. The UDP directly produces cytosine triphosphate. Now you have the C, A, T, C, and G. The confusion is basically from UDP to DTMP. This is the complex pathway. Everything else, if you can come only up to here, then everything can get very easy. Okay, so that's that. Have we all completed the diagram in our notebook? Have we all completed the diagram notebook? Okay, I saw one student who wrote, what about uracil? Uracil is basically needed for RNA, right? And ATCG, these are basically needed for the purines and pyrimidines of DNA. And if we needed uracil, then uracil, it usually comes from the, it usually comes from where? Can anyone tell me where uracil comes from? Uracil, where does the uracil come from? Where? Very good. What happens? How do you convert cytosine to uracil? Very good. D-amino reaction. Now you guys are thinking. Exactly. So when the cytosine is formed, it's deaminated to form the uracil. Are we clear? Okay. Did we complete the diagram? Did we complete this diagram in our notebook? Okay. Good. Very good. Okay. Okay. So can I give you guys 2 minutes? Okay. I'll give you guys exactly 2 minutes, not more than that. Read the diagram once again, and then we will talk about if we have understood the diagram. Can I see the screen again? Okay, of course you can. Let me break it down once again. Ribose 5-phosphate in the presence of PRPP synthetase, that is phosphoribosal pyrophosphate, will form phosphoribosal pyrophosphate. This is phosphoribosal pyrophosphate synthetase, will form PRPP. PRPP, let's choose the easiest route first. That is from phosphoribosal pyrophosphate. You have the direct conversion to ionosine monophosphate. From ionosine monophosphate, you have AMP and GMP. Purine pathway is very easy. Then comes a more complex pathway, that is the pyrimidine pathway. Over here, you have the glutamine, which converts to carbamyl phosphate in the presence of carbamyl phosphate synthetase 2. This 2 is important because it converts the carbon dioxide with glutamine to form CP. And over here, don't forget that this is not a passive reaction, so energy is required. ADP is broken down to form ADP and PI, and a glutamate is released. So after that, we have the carbamyl phosphate in the presence of aspartate. This forms orotic acid. This orotic acid will convert to UMP by binding with PRPP to form uracil monophosphate. And this UMP will convert to diphosphate, right? This diphosphate will be converted to deoxy UDP in the presence of deoxy UDP in the presence of ribonucleotide reductase. I think I forgot to mention this one. Okay, in the presence of ribo UDP will be converted to DUDP in the presence of ribonucleotide reductase. Ribonucleotide reductase. This is also another oncological drug which works over here, which we talked about this in oncology. We'll talk about it once again. DUDP is converted to DUMP. DUMP is converted to DTMP in the presence of thymidylate synthase and a methylation reaction. Okay, so that's that. Okay, can I give you guys how long? 2 minutes? Okay, 2 minutes on the clock. If you're done, please tell me you're done. If you're done, please let me know you're done so that I know that you guys are ready. Okay, anyone else? Okay, very good. Okay, can I get two students who are willing to perform an active participation, not more than two students? Who are the students? Okay. Can we get Dr. Iman and Dr. Jan R? Okay, Dr. Jan. Okay, let's get you guys to unmute yourselves and let's answer the questions over here from the de novo pathway. Can you guys hear my voice? Are you guys ready?
Yes.
Okay, good. So the first question goes out to Dr. Iman. What is the first substrate that is used in the production of purines and pyrimidines?
Ribose 5-phosphate.
Very good. Ribose 5-phosphate. Next question goes out to Dr. Jan. What forms from ribose 5-phosphate?
PRPP.
PRPP. Very good. PRPP. This PRPP forms in the presence of PRPP synthetase. Okay. Then the next one goes that PRPP is converted to which structure for the purine synthesis in this way?
IMP.
Very good. IMP. And over here you have this one and this one. Okay. So what is A for by
AMP.
Dr. Okay. A is AMP by Dr. Iman and B is what from Dr. Jan, please? B.
Guanosine monophosphate.
GMP. Very good. Okay. Very
Is
good.
it
Thank you so much,
necessary
Dr.
to know
Iman.
the steps between IMP and AMP?
IMP is inosine monophosphate and AMP
Yeah,
is
but is it
adenosine
necessary
monophosphate.
to know that we have adenyl succinate and then AMP, or can I just say IMP to
IMP to AMP.
AMP? Okay.
Okay. All right. Next one. Next one is can we get Dr. Mohammed and Dr. Mohammed and Dr. Hussam? Okay. Can we get Dr. Mohammed and Dr. Hussam? Okay. Next one. Next one. Over here you have the pyrimidine
doctor.
pathway. What are the first two structures that is used by Dr. Mohammed first?
Glutamine and CO2.
Glutamine and CO2. Okay, they will combine in the presence of what, Dr. Hussam?
Carbamyl phosphate 2.
2. Okay, this is not an passive pathway. ADP is used over here, so that's that.
Mhm.
And then this is converted to which structure, Dr.
Carbamyl
Mohammed?
phosphate.
Okay, carbamyl phosphate. And next question, Dr. Hussam, which
Yes.
amino acid is used over here?
Aspartate.
Aspartate is used to form which structure over here? Dr.
UMP,
Mohammed?
uracil monophosphate.
No. Dr. Hussam,
Orotic acid.
Okay,
Orotic acid, sorry.
orotic
Orotic
acid.
acid.
Okay, orotic acid will bind with PRPP to form which structure, Dr. Mohammed?
No, UMP.
Which will form UMP. Very good. Next question, Dr. Dr. Hussam. UMP will be used to form which one?
UDB.
In the presence of?
In the presence of.
UMP synthase. UMP?
Okay.
UMP synthase. Okay, thank you so much, Dr. Mohammed and Dr. Hussam. Can we get next one is Dr. Samra Hussain and Dr. Maheshwari. Okay, next one. Next one is UDP is converted to, first of all,
Actually, doctor, UMP synthesis from orotic
Yeah.
acid to UMP, not from UMP to UDP.
right, my apologies, from like over here, exactly. UMP synthesis. We missed out an enzyme. Okay, next one. Next one is from UDP. We have the formation of which structure by Dr. Samra Hussain, please? DUDP. Okay, good. And this occurs in the presence of ribonucleotide reductase. Okay, and which structure is from DUDP, Dr. Maheshwari?
DUMP.
DUMP. Okay, from DUMP. Next one, Dr.
DUTMP.
DTMP.
Yes.
Okay, very good. Okay, can we get the next question directed from Dr. Karbasi, please? DTMP, this formation over here requires an enzyme. Which enzyme is used?
Timidylate synthase.
Imidylate synthase. Okay. Okay. What are we missing out over here? What are we missing out? What do we miss
The,
out?
at the end, you need for the last reaction, you need tetrahydrofolate to
Very good.
dehydrofolate,
Right.
and then again to
Tetrahydrofolate
THF.
to dehydrofolate. Okay. And the enzyme required over here is dehydrofolate reductase. Okay. Dihydrofolate reductase. Okay. So that's that. And we missed out on another one once again. Okay. Dr. Karbasi, would you be kind enough to tell us what's coming over here from the UDP?
CTP.
CTP, very good. Okay. So that's that.
And one
Okay.
question.
Yes.
What is the difference between synthase and synthetase? Sorry for that question, because that confuses me
Basic
sometimes.
formula is not really a lot of difference. It's usually an enzyme which is usually used for the formation of another substrate. That's that.
Okay, so both are the same, synthase and synthetase.
More or less.
So like
More or
more or
less.
less. Okay, thank you.
You're welcome. Okay, so that's that. So are we clear about the de novo pyrimidines and purine synthesis? Yes or no? Okay. Okay. So can we jump into the pathology and the pharma portion of this pathway? Okay. Okay. So the reason why this pathway is important is because there are a lot of drugs which are used in this pathway. Okay, there are a lot of drugs which are used in this pathway to for the purpose of basically oncology. Okay, because most of the drugs are used for oncological pathways, such as we have drugs such as a 6-mercaptolurine, mycophenolate, then we have methotrexate, 5-fluorouracil. So let's find out all the places where these drugs are basically used. Okay, for this one we will use this page over here. Okay, so from over here what happens is this is the reaction which we just studied. The pathway, the first pathway which we studied was from ribose 5-phosphate, you have PRPP. From PRPP you have AMP and GMP. Two drugs which are used on this side. Okay, two drugs which are used on this side is mycophenolate and 6-mercaptolurine. Okay, this is not the last time we will study these drugs. Okay, this is not the last time we will study these drugs. What I will do is that for these drugs we will use the physio subscription, and physio has very good picture mnemonics which we can use to learn about all the informations we need in order to do well in the exam. So this is not the last time we will use, we will this is not the last time we will learn about we will learn about these drugs. But what would happen is there are lots of there are a lot of informations about 6-mercaptolurine and mycophenolate which you need to know more. Okay, but just for now let's get acquainted with the drug. That is the structure. IMP is formed from PRPP, from phosphoribosol pyrophosphate. This pathway over here is inhibited by this drug, that is 6-mercaptolurine and azathioprine. And the pathway from IMP to GMP is inhibited by mycophenolate and ribavirin. Okay. Next one is from over here. The reason why I said that you have to focus on the word 2, carbamyl phosphate synthetase 2, is that we have 2 carbamyl phosphates reactions going on in our system. Number one is carbamyl phosphate synthase 1. Okay, this is used for the urea cycle. This is basically present in the mitochondria, and carbamyl phosphate synthase 2 is present in the cytosol. So the fact that carbamyl phosphate synthase 2 is present in the cytosol and carbamyl phosphate synthase 1 is present in the mitochondria, this is exceptionally high yield for UWorld and NBME both. Okay, so make sure that you guys don't forget this. 2 for cytosol, 1 for mitochondria. 1 is used as the I over here. That's that. Next one is the carbamyl phosphate is converted to orotic acid in the presence of aspartate, and the enzyme which is used is leflunomide. Leflunomide, what this enzyme does is this enzyme, um, it inhibits a very high yield enzyme that is dihydroorotate dehydrogenase. This enzyme is basically not present over here. Okay, and leflunomide will inhibit this enzyme, and what this will do is leflunomide will prevent the formation of orotic acid, and as a result the carbon malphosphate will stay as it is. So leflunomide will do that by preventing dihydroorotate dehydrogenase. And then the next one is 5-fluoro. I'm sorry. The next one is you have the orotic acid, which will bind with a PRPP. The PRPP with orotic acid in the presence of UMP synthetase will form UMP. This UMP synthetase, this can be deficient in a very high yield disease or pathology, which is known as orotic aciduria. In orotic aciduria, what you have is you have UMP synthetase. You have UMP synthase, which is deficient. When UMP synthase is deficient, this orotic acid they cannot convert to UMP, and you have orotic aciduria. You have orotic aciduria. Where do you guys remember another reaction where we have orotic aciduria? Where do we get orotic aciduria? Any other reaction where we get orotic aciduria? Anyone, guys? Do you guys remember orotic aciduria? Where? Where do we get orotic aciduria? Urea cycle. Okay. So when we will study urea cycle, okay, ornithine transcarbamylase. Okay. Okay. So orotic aciduria, it also happens over here because if there's a deficiency of the enzyme UMP synthetase, then you can get orotic aciduria. This orotic aciduria is converted, this orotic acid is converted to UMP, and UMP is converted to UDP, UDP to DUDP. This conversion over here requires an enzyme that is ribonucleotide reductase. Ribonucleotide reductase is inhibited by hydroxyurea, and we have studied about this before. To make things easier, we're going to form a table over here for the drugs. Okay, so no problem. What you have is you have ribonucleotide reductase for hydroxyurea, and then you have thymidylate synthase for 5-fluorouracil. Okay, these are the, and then another one is dihydrofolate reductase, that is for methotrexate. And trimethoprim, TMP, or pyrimethamine, but pyrimethamine is not that high yield. Methotrexate and trimethoprim is high yield. This is the small table which you can use, or you can use this one over here. They already have this. I basically do these things because it helps me learn the oncological drugs in one glance. Okay, so that's that. So you can do that too. Then you have DUMP. DUMP to DTMP occurs in the presence of thymidylate synthase, and thymidylate synthase can be inhibited by the drug that is 5-fluorouracil. And we all know that the drug 5-fluorouracil, it comes from which drug? Which drug breaks down to form 5-FU? 5-fluorouracil. Capecitabine. Very good. So 5-fluorouracil and capecitabine. That okay. Then you have dihydrofolate reductase, which is converting dihydrofolate to tetrahydrofolate, and this is inhibited by the drugs methotrexate and trimethoprim. Okay, that's that. Okay, so that's more or less it. Over here, you also have methotrexate, mycophenolate and ribavirin, which is inhibiting IMP dehydrogenase. That is also another high yield enzyme over here. But to be honest, it's not that high yield. If you can only remember the names of the drugs, that will be enough. Okay, that's that. This is basically all the things that you need to know. And there's only one pathology over here. The one pathology is orotic aciduria, which is taking place because of the absence of UMP synthetase. Another small hidden answer over here, or a hidden question over here, is that pyrimidines, they will require aspartate, and purines will require glycine and glutamate. Okay. require glycine, glutamate, and aspartate too. So over here they use all the 3 amino acid, and over here they only use 1 amino acid. Okay, are we clear about this? How can we memorize all of this? You don't have to memorize these things right now, because when we study, that's what I said. Just learn to get acquainted with the names of the drugs. Yes, learn to get acquainted with the names of the drugs, because later when we use physio to learn the drugs, the physio will help us with the picture mnemonics, and we can use those picture mnemonics to learn about all the things of those drugs. Okay, we clear about this? I do not want you guys to get intimidated by the drugs right now. Try to learn the pathway. Learn the pathway only. When it comes to learning the drugs, we will learn the drugs by using the picture mnemonics from physio. Are we clear? Okay. Repeat about the AA, please. Amino acid, basically purine pathway uses all 3. Pyrimidine pathway uses only 1, that is aspartate. Are we clear? Are we clear about this? Let me show you how we will learn about the drugs. Okay. Let me show you how we will learn about the drugs. Okay, so what you have over here are the drugs which are used. Okay, 1 second. Wait. Over here, you have the picture mnemonics for the drugs. That is, let's see where they are. There we go. So, for example, we just learned about mycophenolate and ribavirin, how mycophenolate is used for it, the fact that it prevents the conversion of IMP to GMP by inhibiting inosine monophosphate dehydrogenase. So this picture mnemonic over here, mycophenolate, this picture mnemonic, this will help us identify all the informations we need to learn about mycophenolate. For example, myphenyl seeds for mycophenolate, and then you guys know exactly how this works. So are we clear about how we will learn the drugs? Yes or no? Yes. Okay. Okay. So we are there. Then we will learn about the drugs. So do we have anything to be worried about? No, we have nothing to be worried about. You do not have to learn the drugs right now. Learn the pathway first. Okay. Okay. Can we move on to the next page? Okay, good. Let's move on to the next page. The next page is basically purine salvage pathway. Purine salvage pathway. Okay, so purine salvage pathway is basically the pathway where we use all the structures that are produced in the process of metabolism, and we use this to restructure the purines back. Okay, we use this to restructure the purines back. So what's happening over here is in the purine pathway, we try to form adenine and guanine, right? So because purines are A and G, so our goal is to make adenine and guanine back from the process of metabolism. So let's take this into another blank page over here. Is everyone ready with their notebooks so that we can start another pathway? Is everyone ready with their notebook? We can start another pathway. Okay. Okay, very good. Let's begin with the purine salvage pathway. Okay. Purine salvage pathway. Okay, purine salvage pathway over here. Okay, the first thing that we need once again, is ribose 5-phosphate. The first thing that we need is ribose 5-phosphate. Ribose 5-phosphate is converted. Ribose 5-phosphate is converted with the help of the enzyme PRPP. It was converted to PRPP synthetase. It was converted to PRPP, and then PRPP was converted to IMP. So what happens is ribose 5-phosphate straight away forms IMP. IMP is inosine monophosphate. This inosine monophosphate, okay, our goal is to make sure that we can form adenine and guanine from the IMP, right? Adenine and guanine from the IMP. And the easiest way that we can form the adenine and guanine from the IMP is basically IMP can be directly converted to AMP and GMP. GMP. And this process can go both ways. Okay. This process can go both ways. Next one is that IMP can be converted. Inosine monophosphate can be converted to another enzyme that is, to another structure that is inosine. Okay, inosine is converted. Okay, inosine is converted to hypoxanthine. Hypoxanthine. Okay, let's wait up to here. Let's come back to this one. That is IMP was being converted to AMP and GMP. AMP and GMP. This GMP over here, this is converted to guanosine. GMP was converted to guanosine, and guanosine was converted to guanine. Okay. And what makes this pathway a salvage pathway, meaning that we are reforming these structures again and again, so that salvage, basically the word salvage, this means that de novo is the word which we use to use the structures present to form the initial pathway of the AMP and the GMP. But salvage is basically we are reusing these structures to form guanine and adenine, so that we never run out of adenine and guanine. Okay, so for example, let's say you do not have enough ribose 5-phosphate. Does that mean that your DNA will stop replicating? The answer is no, because there will be salvage pathways which are using these structures to form and complete these processes again and again. Okay, before we jump into the salvage, let's complete the AMP pathway. Okay, AMP is basically what's happening over here is that adenine, which was formed from the de novo pathway, this adenine, okay, this is helping in the formation of AMP2. Okay, try to realize how GMP is forming guanine by this way, but adenine is actually helping in the formation of AMP. Okay, so what happens is this AMP can now be converted to adenosine. Okay, adenosine can be converted to inosine by, okay, wait 1 second before we link up the pathways. So right now this is what the pathway is. Now is the salvage part. Salvage is basically, now what we have to do is we have to link the pathways. One pathway has to be linked to another pathway, basically making sure that if any one of them decreases, another pathway can go and help form these structures, so that in cases of an emergency or any reason why, when we do not have enough substrate, another process can go and compensate this process. This is known as the salvage. So the first salvage that we want to talk about is this one. Guanine will salvage back into GMP. Guanine will salvage back into GMP, and hypoxanthine will salvage back into IMP by the process of a very, very high yield enzyme known as hypoxanthine, guanine phosphoribosal PRT. Hypoxanthine guanine phosphoribosal and phosphorybosyl. I've really forgot the last one. Let me just see really quick. Hypoxanthine guanine phosphoribosal. Apologies, transferase. I'm not sure if it was translate. Okay, hypoxanthine guanine phosphoribosal transferase. So AGPRT. AGPRT. And what happens over here is that, over here, this is one salvage. Okay, another salvage is this one. That is from adenosine. You have the formation of inosine, and this pathway uses adenosine deaminase. Adenosine deaminase. Adenosine deaminase. Okay, this is another salvage. Okay, this is basically what the salvage is. That AGPRT and adenosine deaminase. Okay, that's that. Now there's also another thing which we can do. We can keep the pathway up to here, okay, and it will not stay incomplete. Okay, but if we have to complete the pathway, then if we complete it, then if you guys remember from previous lecture that if you have hypoxanthine can be converted to xanthine in the presence of xanthine oxidase. Xanthine can be converted to uric acid. This is basically the information that DNA is broken down to uric acid. That's why when we have a lot of meat products that's why when we have a lot of proteins, or for example beef and chicken, which is high in protein, and protein it contains a lot of nucleotides and DNA structures. Have you guys ever heard a doctor advise to a patient who has high uric acid levels to stay away from meat? Yes or no? Yes. Okay. This is the reason why, because all those DNA structures and proteins in the meat that is broken down to add to the uric acid pool. So this increases the patient's uric acid because the purines are broken down to form the uric acid. Xanthine is converted to uric acid in the presence of the enzyme xanthine oxidase, and this uric acid is converted to form allantoin. This uric acid is converted to form allantoin. Okay, and this allantoin is the excreted product. It's excreted in the urine. Okay, and guanine. Do you remember how we said that good Christmas? Do you guys remember good Christmas? Yes or no? Meaning that if you remove the amino group from guanine can also form xanthine by the process of deamination. Okay. Okay. So right now, let's say that for some reason you do not have enough guanine. Okay, you do not have enough guanine. What would happen is IMP can be converted to GMP to guanosine, and guanosine can be converted to guanine. But what if you do not have enough IMP either? Okay, but you have ribose. What would happen is what if you don't have enough AMP either? So if you do not have enough AMP, then this pathway will go. That is, the GMP will be converted to IMP. IMP will be converted to AMP. Then once again, if you do not have enough inosine, and we know that inosine is required to form hypoxanthine. Hypoxanthine can be converted back to IMP, and then this pathway can occur. So in order for this to occur, this process, that is AMP, can be converted to adenosine. Adenosine can be converted to inosine. And do you realize the fact that I just mentioned that there are multiple ways that this pathway can be completed just for the salvage pathway? So there are two salvages, that is hypoxanthine guanine phosphoribosal transferase pathway and adenosine deaminase pathway. Okay. Now, do you guys realize the importance for the salvage pathways? Why we need salvage pathway? Salvage means saving. Saving. You want to form the things. For example, if you do not have enough substrate to form the things de novo, then you can use the salvage pathway. Okay. Are we clear about this? Are we clear about this? AGPRT plus PRPP. Let me see if I missed out on anything. AGPRT plus PRPP. Basically, guanine can be converted to GMP. Hypoxanthine can be converted to IMP in the presence of AGPRT. But sometimes they both can use AGPRT to form PRPP. This is not that high yield. That's why I didn't use it over here. This causes a lot of confusion. So that's that. Okay. Are we clear? Have you guys understood the term salvage? Why they use the word salvage? Okay. Salvage basically meaning if something is absent over here, a pathway can take over and form the substrate once again. If they cannot form the substrate by the help of de novo synthesis, then salvage pathway can come and save them. Okay. Salvage means saving. Let's do a quick review. First of all, you have ribose 5-phosphate, which is converted to IMP. IMP is converted to inosine. Inosine is converted to hypoxanthine. That is the one pathway straight. Then we know from previous knowledge that IMP is converted to GMP and AMP. GMP to guanosine, guanosine to guanine, and AMP is formed with the help of adenine. Now, the salvage pathways are two. That is, guanine can be converted back to GMP, and for guanine to do that, hypoxanthine has to be converted to IMP, and this occurs in the presence of hypoxanthine guanine phosphorybosyl transferase. Okay, and over here, adenine, in the presence of, I think another enzyme was over here. Okay, another enzyme which is not that very high yield, that is APRT. Okay, adenosine phosphorybosyl transferase, or APRT, is converted to AMP, AMP to adenosine, adenosine to adenosine deaminase, forms inosine, inosine to hypoxanthine, and then this reaction keeps on happening over and over again. And then the last one is the last final metabolic products is that hypoxanthine can be converted to xanthine in the presence of xanthine oxidase. Xanthine oxidase can be formed into uric acid in the presence of xanthine oxidase once again, and uric acid is converted to allantoin. Okay. Are we clear? Okay. Can I give you guys 2 minutes? I'll give you guys 2 minutes. Okay. Yes. Dr. Karbasi, yes. Okay. Okay, is everyone ready? Okay. Okay, you know what I will do? Let me just so that everyone can participate. Keep on, okay. Take some more time, try to learn the purine salvage pathway, and let me see the best way I can test you guys. Okay. Best way I can test you guys. Okay. Don't focus on this right now. Focus on the salvage pathways. Okay. Okay. So, okay. So what I need you guys to do is I need you guys to fill in the blanks with the letters. First of all, let's talk about A. Which one is A? Which one is A? A is IMP. A is IMP. Very good. Which one is B? PRPP. Which one is C? Very good. A and B. Which one is D? Very good. Okay. Which one is G? Okay. Which one is E? Okay. Which one is F? Okay. Which one is okay? Good. So basically, you guys get the hang of it. Now, are you guys sure and confident that when you guys receive questions from Pure and Salvage Pathways, if they ask you to identify an enzyme, you can identify with absolute perfection? Okay. How confident are we with the Pure and Salvage Pathway? Confident. Okay, very good. Okay. Okay, very confident. Very good to know. Okay. So before we continue further, do you guys want to take a break? Or later? Okay, because I need you guys to be on your A game over here. Okay. Okay, 15 minutes. All right, 15 minutes. Okay, go ahead. So I'll see you guys in 15 minutes. Okay. Is everyone back from their break? Okay, good. Okay. All right. So let's go back to our lecture. Okay. So this is where we were. We were studying the purine and pyrimidine salvage pathway. Okay. And that's that. Right now, we will talk about some of the pathologies associated with this pathway. The first pathology that I want to talk about is, once again, what was the enzyme over here for G? What was the enzyme for G? HGPRT. AGPRT, meaning hypoxanthine guanine phosphoribosyltransferase transferase. If, for example, you have a deficiency of the enzyme HGPRT, you have a very high yield pathology that is known as Leach-Nyhan syndrome. Leach-Nyhan syndrome is a defect in your purine salvage pathway when AGPRT is absent. So what happens is, if you have a defect in the purine salvage when AGPRT is absent, AGPRT is used for the conversion of hypoxanthine to IMP, this conversion over here. And also it's used for the conversion of guanine to GMP. So when this doesn't happen, this results in excess uric acid formation. Basically, if this pathway cannot go this way, right? This pathway goes this way. If this pathway keeps on going this way, then hypoxanthine is converted to uric acid. So this results in excessive uric acid production and de novo purine synthesis. So what happens is, these patients, what happens is they have a certain amount of clinical features. Let me have a blank page over here so that we can talk about the clinical features. Clinical features are important for a pathology, basically, because this is the way the questions will come in your exams, right? The questions will appear according to the clinical features that is present in the textbook. So we're talking about Leach-Nyhan syndrome. Okay. You have the clinical features of Leach-Nyhan. The clinical features of Leach-Nyhan is that these patients, they will have excessive uric acid, and they will have, and it's an X-linked recessive disease. So first of all, this is an X-linked recessive disease. Leach-Nyhan, it's an X-linked recessive disease. What happens over here is that these patients, they are intellectually disabled. They're not very intelligent. Okay. Then these patients, they have self-mutilation or self-mutilating behavior. One of the most common problems the mother will come and complain about their child is that their child is biting away their lips. The child will try to self-mutilate. The word mutilate means harm, and they will try to harm themselves. And the way they will try to harm themselves is that they will try to bite off their lips. The way I like to think about it is that over here you have a problem with purine formation, right? And because AGPRT is not there, so your body keeps on looking for purine products, meat products. So the first protein that is right in front of your mouth are your lips. So the young child or the patient, they try to bite away their lips. Patients of Leach-Nyhan syndrome, they have a very scary appearance. Okay, they have a very scary appearance. So this is how Leach-Nyhan syndrome patients usually present. They bite away their lips, like this one. They try to look for proteins. The first protein is their lips, so they try to bite away their lips. Pretty scary. So that's that self-mutilating behavior. Next one is that these patients, they have what they have is they have extreme aggression. They're always very aggressive. They're not calm. Okay, they're not calm. So these are the three things that the mother will complain, and this will be present in your question. The signs that you will see as a physician, is that these patients, when you do their comprehensive metabolic panels and you see their uric acid level, you will find that these patients, they are they have a hyperuric acid level, meaning that they have hyperuricemia. Uricemia. Okay. Next one is with uric acid comes gout. They can also have signs symptoms of gout, right? Then in CBC they can have macrocytosis because there is impairment of proper DNA formation. As a result, RBCs are larger, meaning MCVs will be higher. Okay, another one that you will find over here is that you will find when you try to ask the mother that when you change your baby's diaper, what do you find? The mother will tell you that there is an orange color appearance of the diaper. This orange appearance are sodium urate crystals. This excessive uric acid that is produced will bind with the sodium to form an orange salt known as sodium urate. So sodium urate will form an orange colored diaper, orange diaper. Okay. Okay. Are we clear about this? Are we clear about the clinical features and the findings? Okay. Good. Very good. How will you treat these patients? These patients, unfortunately, the only form of treatment that we can give them is we can stop the pathway over here. Although it still does not treat the self-mutilating behavior of the patient, but at least what it will do is it will prevent the formation of the uric acid. So you can prescribe allopurinol and febuxostat to inhibit the enzyme xanthine oxidase. So treatment is basically allopurinol, which is the first line. The second line is febuxostat. Okay. Leach-Nyhan syndrome. This is Leach-Nyhan syndrome. Once again, the enzyme that is deficient is HGPRT. Are we clear about this? HGPRT, Leach-Nyhan syndrome. Okay, next time, if you guys receive a question about HGPRT and Leach-Nyhan syndrome, look out for a young child, aggressive, biting away their lips with painful joints due to gout, then extremely aggressive, orange colored diaper. All these are signs and symptoms of Leach-Nyhan syndrome. Another one is also a very high yield disease. That is, if HGPRT is absent, what was another enzyme that we used for the salvage pathway? Who remembers what was another enzyme that was used for the salvage pathway, which the one which we drew in blue? Adenine D-minus, very good. Adenosine D-minus. Adenosine D-minus, if adenosine D-minus is not there, you have another high yield disease. This disease is known as a very unfortunate disease because the prognosis of this disease is not that long. The mortality is very high. This disease is known as severe combined immunodeficiency syndrome, or SCID. SCID. And adenosine D-minase, if that is not there, adenosine D-minase is the enzyme that is deficient. And this is autosomal recessive. Okay, adenosine D-minase is responsible, right? Adenosine D-minase over here it is responsible for the degradation of adenosine to inosine, right? So if we do not have enough, if we do not have this enough enzyme adenosine D-minase, you have increased amount of adenosine. This increased adenosine is very toxic in the fact that this increased adenosine, what will happen is this will result in decrease in ribonucleotide reductase activity, ribonucleotide reductase activity. And this increased adenosine, this is extremely lymphotoxic, meaning that this will kill your T lymphocytes and B lymphocyte, but especially your T lymphocyte. So what this will do is this will cause T lymphocyte deficiency. So this will attack your T and Bs, but mostly T. So the patients have severely compromised immunodeficiency syndrome. Okay, severely compromised immunodeficiency syndrome. Are we clear about these two diseases, adenosine D-minase and AGPRT? Adenosine D-minase deficiency, if that is not there, there will be increase in adenosine. Increased adenosine, if adenosine deaminase, if that is not here, then there will be increased amount of adenosine. If there's increased amount of adenosine, what will happen is that there will be extreme formation of DATP, increased formation of DATP. DATP is DATP. What it will do is it will result in decreasing in the ribonucleotide reductase, because everything will go to this way, DATP. So this pathway will not be there, and ribonucleotide reductase will work less. If there is decreased activity of the ribonucleotide reductase, then this is extremely lymphotoxic, and also adenosine itself is extremely lymphotoxic. But the fact that ribonucleotide reductase activity is decreased, it's not very high yield for adenosine deaminase. Remembering that adenosine deaminase deficiency forms SCID, severe combined immunodeficiency, is enough. That is enough knowledge for step 1. Okay. Okay. Are we clear about this? Can we move on? Are we clear about these two diseases? Okay. All right. Next one. Next one is your genetic code features. Okay. Next one is genetic code features. Genetic code features, especially that. Do you guys know about how you need 3 codes, right, to form? How you need three genetic code. I can't. Okay, we have a question from Dr. Karbasi. I can't see DATP. DATP is usually not formed, but when you have excess amount of adenosine, then DATP is formed. If DATP is formed, it decreases the ribonucleotide reductase activity, and that is lymphotoxic. That's that. Okay. It's not usually formed. Okay. My question is, have you guys heard about the fact how 3 codons are required to form give out one genetic information, right? Yes or no? Okay. And each codon which contains each codon, for example, each codon can specify only one amino acid, unambiguous. The fact that each codon is specific for one type of amino acid, this is known as an unambiguous feature. Unambiguous, the word unambiguous meaning that it's that basically it means that it's a given. It's a given, meaning that it is very well known that each codon, it specifies for only one amino acid. So if you have 3 codons for like this, for example, methionine, the codons are AUG, right? No matter what, every time AUG, if it forms in a DNA structure, then this will give rise to methionine, which is the amino acid. So that's what it means. It is very unambiguous that AUG will form methionine. That's that. Next one. Next one is degenerate. This one is high yield, extremely high yield, degenerate codon. Extremely high yield. What is a degenerate? Basically, most amino acids are coded by multiple codons. Most amino acids, they're coded by multiple codons. The wobble is basically the 3rd position of the codon. The codons that differ in the 3rd may code for the same tRNA acid. So basically, if you were to change the 3rd place, for example, there is, for example, it's GAG. This last G is the wobble position. The first 2 are important for deciding what amino acid it will be. After the decision is made, even if you change the last one, the G, it will still not matter. That amino acid will still be produced. This question is asked a lot in your NBME, especially when they ask you that which type of genetic feature, if removed, can also form the same amino acid regardless. The answer is the wobble codon, the wobble position, meaning wobble basically means it does not matter. The 3rd position, it does not matter. Okay. Specific base pair is usually required only in the first 2 nucleotide positions of the mRNA codon. Okay. That's that. So the 3rd position is not that. We used to joke around in our medical school how when we wouldn't care about someone, we would say that they're the wobble. Okay, that if there was some professor or some type of a teacher that we really didn't like and we really wanted to avoid them, and this keeps on happening a lot in medical school. I'm sure you guys have your likes and dislikes about your teachers. We usually used to say that the teacher which we didn't like was the wobble teacher, that he or she did not matter. Okay, so you guys can also use this to remember that wobble means it does not matter. The position, it does not matter. Okay, so the wobble position. Next one is commaless or non-overlapping, meaning that you read from a fixed starting point as a continuous sequence of bases. Okay, so that's not very high yield. So you start reading from one point. For example, you have A, U, G, A, G, then U again, A again, right? You start reading from here. This is one codon, this is another codon, and then you have some more, right? So the fact that you start from this starting point, this is the commaless non-overlapping feature of the genetic code. That's that. The universal is basically the genetic code is conserved throughout evolution, basically meaning that the code for mitochondria, for example, let's assume that you have a codon for mitochondria. Let's say the codon for mitochondria is GAG. Once again, this is just an assumption. The fact that GAG will code for mitochondria, this is universal, meaning that this is how it has been in the humans for billions and billions of years. That is, throughout evolution, everything has changed, but the fact that these three coded for mitochondria is still the same. So this feature is known as a universal feature. Not high yield, not important. The only high yield thing from over here is the wobble, meaning the part of the codon or the third position of the codon, which does not matter at all. Okay, are we clear about this? The type of questions you would receive in NBME is basically they will ask you which position of the genetic code is not important for coding for the same amino acid, and the answer is the third position or the wobble position. Okay, AUG is the start codon. Yes, augment. Okay. Okay, let's discuss this since you asked about AUG. Okay, AUG is used for the word augmentation, meaning AUG is methionine. That augmentation meaning start or the beginning. So methionine. And the stop codons are you are away, you go away, you are gone. You are away, you go away, and you are gone. These are the stop codons. Okay, are we clear about this? Okay. Did you guys know about this before? I'm sure you guys did, right? The start codons and the stop codons. You are away, you go away, you are gone, right? Yes, very good. Okay, that's good. Now comes the mountain of information over here. That is DNA replication. Okay, how many of us are familiar with DNA replication? How many of us are familiar with DNA replication? Very good. Okay, very good. Okay, so what I will do, okay, so what I will do is before we jump into this whole process of enzymes, what's happening over here, can I give a very brief, small introduction to DNA replication before we jump into first aid? Okay, we will give a introduction to the DNA replication before we jump into first aid, so that for all the students who are not well accustomed to what DNA replication is, okay, basically there are two processes. One is DNA replication, another one is protein synthesis. Do not mix those both up. DNA replication is that DNA, it is replicated to form another DNA. Okay, DNA replication and is formed to it is basically formed to DNA. I mean, this whole process, this occurs because because in DNA replication, you had the process of replication has to occur for another DNA to form. That's that. Nothing more, nothing less. So if we have a DNA, right, we know that the DNA they have, for example, like this, they have the 3', okay, they have 3' end, and they have the 5' end, and then over here they have the 5' end, they have the 3' end. Okay, what do we mean by the word primes? What do we mean by 3' and 5'? Does anyone know? What do we mean by the word 3', 5'? Exactly. 3' and 5' basically mean that at the third position and at the fifth position, you have the attachment of the carbon atom. You have the attachment of the carbon atom. So let's say that this was the structure over here. Okay, so you have 1, 2, 3, 4, 5, 6. This is the base structure. Over here, if you have the attachment of the carbon atom, this is the 3'. And you have the attachment of the carbon atom over here, this is the 5'. That's that. So that's basically what we mean. Okay, so basically, if the first process of DNA replication, okay, the first process of DNA replication is that first, before the DNA replicates, it has to be divided. DNA has the 3' end and the 5' end, right? How we just said before. It has to be separated and unwind. It has to be separated and unwind into big structures over here. Okay, this is basically the 3' end. This is the 5' end of the DNA. Okay, the process, the enzyme that is used for unwinding, you know how the DNA is super coiled like this, right? This has to be unwinded, unwinded in order for the replication to occur. The enzyme that is responsible for the unwinding of this structure over here, this enzyme is known as DNA helicase. DNA helicase. So DNA helicase is basically the enzyme that is used for the unwinding of the DNA. Okay, right now you have the both strands of the DNA, that is the 3' end and the 5' end of the DNA, and it is unwound by the DNA helicase. This unwinding of the DNA helicase, this is known as the replication fork. What do we mean by the replication fork? This means that if anything were to get replicated, if it continues in this direction towards the replication fork, it will be a smooth process. If it goes away from the replication fork, it will not be a smooth process, meaning that this is basically the point from which the replication, this is the point at which the replication should end, right? So anything this way is not good. Anything this way is good, okay? It's a bit silly, I know, but this is only for your understanding, for the fact that because I want you to understand what the leading strand is and what the lagging strand is. Okay. Now, what happens over here, if this is DNA helicase and if this is the replication fork, okay, first of all, what happens is you have, so if this is the DNA, the replicated thing that should form from the DNA is RNA, okay? So from over here at first, what you have is you have the origins of replication. Origin of replication is basically what happens is you have primers. Primers are DNA primers. DNA primers are basically meaning that preformed RNA structures. These are preformed. So what happens is this preformed RNA, they go and they bind over here. They bind over here. So what this does is we'd like to think about primers as a boost. We like to think about primers as a boost, and meaning that the primers will boost or begin the process of DNA replication. Basically, before we do anything, for example, right before we start running, if someone comes and tells us and motivates us and boosts us up that, okay, if you run really well, you're going to win, and that the motivator, right? So DNA primers are the motivators or the boost ups. So these are primers. So DNA primers, they come and they bind to this part where the DNA primers, they come and they bind. This is, I mean, the RNA primers, they come and they bind. This RNA primers, this part is known as origin of replication. Are we clear about these informations? Yes or no? Okay, good. What happens after the RNA primers, they bind, the enzyme that comes, the next enzyme that comes is DNA polymerase 3. DNA polymerase 3. DNA polymerase 3, what this does is this keeps on forming the RNA, right? RNA, and it keeps on moving in this direction. So we can say that DNA polymerase 3, DNA polymerase 3, they have, so this is the first thing, that is, first of all, if this is the 3 prime and the DNA polymerase 3 comes over here, then can we say that the DNA polymerase 3, this added an RNA primer, and from the primer, DNA polymerase 3 kept on moving in this direction. If this is the 3 prime, then this is the 5 prime at which the DNA polymerase 3 came and it started beginning the replication fork. Yes or no? Can we say this, that the DNA polymerase 3, it came and it bound at 3 prime. If they have to attach another substance, that has to be at the 5 prime. That is the exact opposite, right? Okay, so DNA polymerase 3 came and it attached the RNAs all the way, right? And it kept on moving this way until it reached the replication fork. So it's a continuous strand. So can we say that DNA polymerase 3 have 5 prime to 3 prime synthetic activity? Yes or no? 5 prime to 3 synthetic activity, yes or no? Okay. And there's also another thing. My question is there a possibility that when DNA replication is taking place, can there be an addition of a faulty RNA, and this can mess up the whole procedure because there's so much DNA replication going on in the body? Is that possible? Yes or no? Yes or no? Yes. If that's a possibility, then what DNA polymerase 3, what they do is they proofread the DNA. They will read the, so once they finish the replication this way, once they finish the replication all the way, they will come over here, right? They will come over here, and then they will start reading the whole thing that, okay, I have added all these RNA primers. I just want to make sure that these are added in the right sequence. And for example, they find something over here which is not right. Okay. What they will do is they will remove this portion and add another primer. So can we say that they have 5 prime to 3 prime synthetic activity, and for example, this is 3 prime, right? 5 prime to 3 prime synthetic activity, and then they have 3 prime to 5 prime. They have 3 prime to 5 prime exonuclease, meaning that exonuclease is basically the fact that our DNA polymerase 3, they found this faulty RNA over here. They got rid of this portion of the RNA. This getting rid of the fact, this is known as exonuclease activity. So can we say that they have 3 prime to 5 prime exonuclease activity? Yes or no? Can we say this? Yes? Okay. Guys, have you guys understood? If you guys did, then can I get a yes on the chat box or a no on the chat box, please? Okay, good. So this is a very, very high yield piece of information, especially for your NVMe. Okay, that next time if you receive a question and you have all these information over here that they go like, which enzyme has 3 prime to 5 prime exonuclease activity and 5 prime to 3 prime synthetic activity, and they have DNA pole 1, 2, 3, right? RNA pole 1, Now, if you receive these type of questions, are you going to be confident, yes or no, that it's the DNA polymerase? Okay. And is there a possibility that you guys can receive these questions? Have you guys already received these questions in NBMEs? Yes or no? Anyone? Anyone? Yes. Okay. Good. Very good. So you guys know how important this is. Okay. Next one. Next one is the fact that there was a primer here. Do you guys remember the RNA primer that was over here? The fact that the primer is an RNA. Do not forget this. Primers are RNA sequences. We know a lot of students who are not sure whether the primer is DNA or RNA. The primer is an RNA. Now, this primer that was over here, this primer has to be removed. This primer has to be this primer. This this primer. The fact that the primer was here, the enzyme that added this primer is known as primase. My apologies, DNA primase. Okay, primase. This enzyme is known as primase. DNA primase. Okay, and this strand that has just been produced in this way, without any lags, this strand is known as leading strand. This strand is known as leading strand. Okay, now we will talk about the next strand that is over here. That is this one over here. DNA synthesis will always occur in the opposite of 3 prime. So it will occur in this way. But what happens in this one over here, that DNA synthesis, this occurs in the opposite direction. So since this is the replication fork, okay, this is the replication fork. DNA synthesis, if it occurs in the opposite of the replication fork, this occurs at first. What happens once again, that over here you have primase, right? The primase, this comes and this attaches an RNA primer over here. Okay, this attaches an RNA primer, and then you have which enzyme? You have DNA polymerase 3, right, which comes and it starts making the strand. DNA polymerase 3, it starts making the strand. But the thing is, since the fact that DNA polymerase 3 comes and it's going away from the replication fork, this does not happen in a continuous sequence. So what happens is over here, DNA polymerase 3, it moves, and then over here it stops. Okay, and then once again, what happens is the primase, they come, they attach another primer, and then the DNA polymerase 3 comes again, and they start this sequencing. And then again, they stop because it's going away from the replication fork. So it doesn't want to go, because it's easier to go towards this way than to go away, because it's pulling the DNA polymerase 3 towards this side. So since it has to move away, it works in little tad bits. And then once again, the RNA primase come, and then DNA polymerase 3 comes and it completes the lagging strand. So these small fragments, these fragments, they have a certain name. Their names are Okazaki fragments. These fragments are known as Okazaki fragments. Okay, and what happens is after this one, the process is still not done. You have these gaps in between the DNAs, right? You have these gaps. These gaps, they have to be fulfilled, and the enzyme which fulfills this gap is that we have another enzyme. This enzyme goes and it forms a ligation between these gaps by forming a phosphodiesterase bond. This ligazing enzymes, these are known as DNA ligase. These ligazing enzymes are known as DNA ligase. Now it's a complete strand. Once this strand has formed, this strand is known as, this strand is now known as lagging strand. It will lag behind. This is known as lagging strand. Okay, that is basically what this is. Do you have another piece of enzyme? This is known as DNA. This enzyme is basically over here. This enzyme, it stays over here. Okay, this enzyme is known as DNA topoisomerases. DNA topoisomerases are basically so the DNAs, they are very supercoiled. Supercoiled, meaning that the DNAs are coiled very condensely, and a lot of coiling is there. So in order for this DNA to separate, the helicase will separate the strands. But even before helicase comes and separate the strands, topoisomerase will help removing the supercoil. So if we were to block this enzyme, then DNA replication will not occur. And we use a lot of drugs which are used to block topoisomerase 1 and topoisomerase 2. And we have talked about this before, that topoisomerase 1 are irinotecan, topotecan. Topoisomerase 2 are etoposide and teniposide. So those are the drugs. So topoisomerases are the enzymes that are used to prevent DNA supercoiling. And that is your DNA replication. Are we clear about DNA replication? Everyone? Replication fork is nothing but the point from where replication begins. Okay, the point where replication begins. You have another structure over here, that you have these single-stranded binding proteins. Okay, you have these single-stranded binding proteins. So the single-stranded binding proteins, they basically, what they do is, these proteins, they make sure that the DNA does not go back to the coiled up structure. Once they have been unwounded, they make sure that these DNA structures do not go back to the supercoiled structure it was before. And the DNA helicase and the single-stranded binding protein, and the fact that the replication begins from over here, this is known as the replication fork. Okay. We did not discuss DNA polymerase 1. We discussed DNA polymerase 3 only. Okay. Have you understood DNA polymerase 3? DNA polymerase 1, we have not discussed it as of yet because it's not there in eukaryotes. Do you guys know the difference between eukaryotes and prokaryotes? Humans, are they eukaryotes or are they prokaryotes? Okay, good. Dr. Ahmed, what is your confusion? What is your confusion? Please, if you have any question, ask me right now. Okay, repeat about the second part of DNA polymerase. Second part of DNA polymerase. Okay, which part are we talking about over here? Second part of DNA polymerase. Can you please unmute yourself and ask me the question, because it will be faster. I have to keep on moving forward, please. What is your question? Unmute yourself and ask me the question, please.
Doctor, about the primers, you said that it's got in the replication fork, right? And then you said it is inhibited here.
Where?
Yeah, here. Exactly here, where you are. It's inhibited.
Here?
Why it's inhibited and how? Yeah.
It's not inhibited. What happens is RNA primers, they begin the process of replication. But the problem with this part is that when the DNA replicates away from the replication fork, it only occurs in small amounts. It does not occur continuously like this one over here. Because when it goes away from the replication fork, it's harder to replicate. Okay, so what happens is RNA primer comes, DNA polymerase 3 comes, and forms a new RNA sequence, and then it stops. Then again, RNA primer comes, and then DNA polymerase 3 comes again, it replicates, and then it stops. And then again, it keeps on continuing like this. That's what's happening over here. Are we clear?
And what DNA ligase did it do to it? Sorry.
DNA ligase, what they do is after there's formation of the strand, there are pieces of blanks in the middle. So what happened was that over here you had a blank, right? Primers, polymerase 3 forms a new strand, blank. Primers, polymerase 3 forms a new strand, blank. So if we have to complete these sequence, then these blanks have to be fulfilled. This fulfilling is done by another enzyme, just for the lagging strand, not for this one. This enzyme is known as DNA ligase. They bind this one with this one. They just bind this one with this one. Okay, by phosphodiesterase bond. Okay, are we clear? Dr. Mohammed, are we clear? Okay. Next question is from Dr. Garbasi. Topoisomerase, repeat. Topoisomerase is basically nothing. These are basically enzyme which allows the DNA to topoisomerase. Basically, they allow the DNA to remove their supercoil. The DNA is very supercoiled. If you do not remove the supercoil, you cannot separate the DNA. So they allow for the removal of the supercoiling of the DNA. Okay. Telomerase. I did not discuss telomerase yet. I have just discussed links. Okay. Replication fork is basically the point from which replication should begin. Okay, for the leading strand. I mean, should end. Okay, are we clear about this? Telomerase, DNA polymerase 3. These are the things I have not discussed yet. This is just an introduction to the diagram. This is not the actual stuff. We will read about the first aid. We will read it in the first aid right now. Okay, so let's move on to the first aid. Let's see what's there. Okay, so first of all, we have DNA replication. Eukaryotic DNA is more complex than prokaryotes. Prokaryotes are not us. Prokaryotes are basically microorganisms, but uses many enzymes and analogs to those listed below. So there are a lot of similarities and dissimilarities in between eukaryotic and prokaryotic DNA replications. In both prokaryotes and eukaryotes, DNA replication is semi-conservative, meaning that it involves continuous and discontinuous synthesis, and it usually occurs in the 5' to 3' direction. Synthesis always occurs in the 5' to 3' direction. Okay. Right now, origin of replication. This is the first one, that this part is the origin of replication. Origin of replication is basically particular census sequence in a genome where DNA replication begins, maybe single or the multiple. Basically, the point from where RNA primer comes and starts the process of multiplication. This is known as origin of replication. Over here, nothing more, nothing less. For this one, you have only one origin of replication. For this one, you have multiple origins of replication. Okay, that's that. AT-rich sequences are found in the promoters and origins of replication, such as Tata box. This is extremely high yield. Okay, this is extremely high yield for you all. If you're doing a fast review, then you have to understand that promoters, basically origins of replication, they contain high amount of AT, meaning adenosine and thymine sequences, and these are known as TATA box. TATA box. Are we clear about this? Next time, if you hear the word TATA, I want you to think about replication and origins of replication and the promoter regions of replication. Are we clear about this? Where are the promoters? The promoters are not here. Over here, you only have the origins of replications. We'll talk about the promoters later. The promoters are there in the genes. Okay. 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 Next one is replication fork. Replication fork is basically this part over here. Okay. Replication fork is the Y-shaped region along DNA where leading and lagging strands are synthesized. That's that. Nothing more. Not very high yield. Next one is helicase. Helicase unwinds DNA template at replication fork. This is very high yield. If they ask you a question that which enzyme is responsible for the unwinding of the DNA template at the replication fork, the enzyme is helicase. Okay. And I'm pretty sure after reading this, you guys can easily understand. you guys can easily understand the helicase. Okay. If you receive a question about helicase, can you answer them right now? That which enzyme is the helicase? Yes or no? Yes. Okay. Is everyone understanding the lecture? Okay, good. Is this the first time? Do we have any students over here for whom DNA replication today is the first time? Do we have any students over here? Okay. Have you guys? Oh, no problem. Okay, big, no problem. But since being this is the being the fact that this is your first time, have you understood DNA replication? Have you understood the process which I just talked about? Okay. Have any question? Please don't hesitate to ask me right now. Next one. Next one is, so we were talking about the enzyme helicase. Helicase is responsible for DNA unwinding at the replication fork. And if we have a deficiency of the enzyme DNA helicase, we have a very important condition. This is known as Bloom syndrome. Bloom syndrome is responsible for a gene mutation that is BLM. Bloom syndrome, the patients of Bloom syndrome, what they have is, they usually have, let me go over here. Bloom syndrome. How do you identify a patient with Bloom syndrome? I'm surprised how they don't talk a lot about Bloom syndrome on first day. This is the only piece of information they have. I'm not sure if they added anything new. Another one is, if you have a patient with Bloom syndrome, what you have is you have high pitched voice. Then you have long, narrow face. And you have prominent nose and ear. This is Bloom syndrome. What happens over here? Bloom syndrome. Okay. Bloom syndrome. And another one is they can also have short stature. Short stature. Okay. Okay, over here. So what happens is that these patients, they're heavily predisposed to cancer. They're very, very predisposed to cancer. If you do not have a proper functioning of the DNA helicase, can DNA replicate and form new DNAs? Yes or no? Yes or no? Can I get some fast answers, please? If you do not have DNA helicase, can the DNA replication take place? No. So if you have oxygen which is affecting your DNA and your DNA has to be reformed, can it do that? Yes or no? If it does not, if the DNA cannot replicate and move away from the carcinogenic exposure, a single carcinogenic exposure can result in the formation of cancers. So predisposed to carcinoma. These patients are extremely predisposed to carcinoma. Okay, let me show you a picture of Bloom syndrome. Bloom syndrome. Okay, it's a rare disease. It's a very, very rare disease. They also have these facial structures and facial features, and that's that. They have distinctive facial features, right? Predisposed to cancer, right? And that's that. Okay, all right. Next one. Next one that we want to go to is single-stranded binding proteins. Single-stranded binding proteins are these ones over here. They prevent the DNA to go back in this way, because if the DNA goes back in this way, then DNA replicate cannot occur. So that's that. If this is also a high yield question, which structures are responsible for prevention of DNA re-annealing? And the structures are single-stranded binding proteins. Next one is DNA topoisomerase 2. DNA topoisomerase 2 are the ones over here. They create a single or double-stranded break in the helix, and they remove the supercoils. this supercoiling of the DNA. If it's not removed, then the DNA cannot separate, right? So DNA topoisomerase will remove the helix to remove the supercoil. So that's that. And then once again, you have the drugs. Okay, I'm sure we talked about this once, but I love talking about this. I'm going to talk about this once again. You have topoisomerase 1. So you have irinotecan and topotecan. So these are the two drugs which goes and stops topoisomerase 1. Drugs which stops topoisomerase 2 are atoposide and 10-2. Okay, atoposide and tenoposide. That's that. So atoposide and tenoposide, these are the drugs which will go and stop topoisomerase 2. Then in prokaryotes, you have fluoroquinolones, and which will inhibit topoisomerase 2. And in prokaryotes, they have another topoisomerase, that is topoisomerase 4, that is inhibited by DNA gyrase inhibitors. And we will talk about this in microbiology. Okay, that's that. Primates are the enzymes which are required to add the RNA primers. If there were no addition of RNA primers, the DNA replication will not begin. So that's that. So RNA primers are added by the help of the enzyme. Primates. Keep in mind that questions are usually going to come from this side. I mean, the questions are usually going to come from this side, and the answers are on this side. So if you have to test yourself, cover this part with your hand, read this out, and see whether you remember these names or not. Okay, that's that. DNA polymerase 3, one of the highest yield that DNA polymerase 3, they are present in prokaryotes. They elongate the leading strand by adding deoxynucleotides to the 3' end. They will elongate the lagging strand until it reaches the primer of the preceding fragment. DNA polymerase 3 has 5' to 3' synthesis and 3' to 5' exonuclease activity. We talked about this a little bit back. Drugs that will block DNA replication often have a modified 3' hydroxyl group, thereby preventing the addition of next nucleotide. So they want to prevent chain termination. So we can use drugs that can add the 3' end and hydroxyl group, and that can prevent the addition of the next nucleotide. Okay, are we clear about this? Does anyone have any question? What are your questions? Does anyone have any questions? Indicated for what? What is indicated for what? It's been tested in USMLE. Okay, I'm not. Okay. Can you please rephrase your question? Last two symptoms of Bloom syndrome. Last two symptoms of Bloom syndromes are basically short stature, prominent jaws, and then predisposed to carcinomas. Okay, that's that. Next one, re-annealing means binding. Yes, re-annealing means binding. Bloom syndrome, it has been tested. Yes, it's tested in NBMEs. Okay. Are we clear? Indication for topoisomerase inhibitors, they are anti-cancer drugs. Etoposide, tenoposide, irinotecan, and topotecan are anti-cancer drugs. Any other question? Okay, next one. Next one is DNA polymerase 1. DNA polymerase 1, they are present in the prokaryotes. They will degrade the RNA primer and replace it with DNA. So the primer, which was over here, this could have been broken down by DNA polymerase 1. DNA polymerase 1 will break down the RNA primer, and they will replace it with DNA. That's that. And this has the same function as DNA polymerase 3. Also, the fact that they can excise the RNA primers in 5' to 3' exonuclease. Okay. DNA polymerase 3, do they have 3' to 5' synthesis or 3' to 5' exonuclease activity? Anyone? Synthetic activity. Synthetic activity is from side to which side? 5' to 3' synthetic and 3' to 5' exonuclease. So please write this down in your book over here or with a post-it note. DNA polymerase 3 has 5' to 3' exonuclease, and DNA polymerase 1 have 3' to 5', 3' to wait, they also have, my apologies, DNA polymerase 1, my apology, I think I kind of mixed up. DNA polymerase 3 has 3' to 5' exonuclease. My apologies, so sorry. 3' to 5' exonuclease, and DNA polymerase 1 have 5' to 3' exonuclease activity. Okay. Okay. Yes, I mixed it up. My apologies. There's so much going on in my mind right now. Okay, my apologies. I'm really sorry. Should not have mixed it up, but that's that. Okay. Are we clear about this? Okay. Is there a mistake in state? Why will there be a mistake about DNA polymerase 3? What is the mistake? What is the mistake?
Sorry, because it's written in DNA polymerase 3 prokaryotes only. But I mean, we just discussed about eukaryotes, right?
Right. We just discussed about eukaryotes, but the thing is, DNA polymerase 3, although it says that it's just there on prokaryotes, it's not a mistake per se. But the thing is, in eukaryotes, it's also used to some extent, but in prokaryotes, it's used mainly. It's used mainly in prokaryotes. In eukaryotes, DNA polymerase 3, instead of DNA polymerase 3, in eukaryotes, sometimes you do not even need the enzyme DNA polymerase, because once you have the primers, when the primers are added, the synthesis can keep on happening in the leading and the lagging strand. So that's that.
So are we right now talking about the prokaryotes, or then I got confused?
Oh, which one? This one?
Yeah, the space.
here?
Are we talking about the prokaryotes, or I thought about the eukaryotes?
No, we were talking about the eukaryotes. We were talking about the eukaryotes in the sense that I was discussing this diagram over here. But in eukaryotes, you have the DNA polymerase 3, which are working. But the thing is, in DNA polymerase 3 in eukaryotes, they can work, but at the same time, they can also the leading strand can also form without the presence of the DNA polymerase 3 when there's addition of the primers, so they can easily start formation over it. But in prokaryotes,
Okay.
DNA polymerase 3
Yeah.
is it's needed regardless.
Okay, but still we talk about the eukaryotes. Okay, I got it. Okay.
Is everyone clear about this? Okay, is everyone clear? Is everyone clear about this? Do we have any questions? Guys, do we have any questions? Can we move on? Okay. Okay, so which enzyme has proofreading abilities? That enzyme wants to go and check whether it has added the nucleotides in the proper way or not. Okay, that DNA polymerase 3 has proofreading abilities. Please write this down over here. Unfortunately, I do not have enough space on my page. Okay, write it down over here. DNA polymerase 3 has proofreading abilities. Okay, next one is DNA ligase. DNA ligase will go and catalyze the formation of the phosphodiesterase bond. That is over here. They will come and then they will bind these two structures together. Okay, that's that. Now we have come to a very important structure, that is, a very important enzyme, that is telomerase. Telomerase are basically, they're present in eukaryotes. Basically, this is a reverse transcriptase, that is an RNA-dependent DNA polymerase that will add the DNA to the 3' end of the chromosomes to avoid loss of genetic materials. What happens is they will come and add or tag, meaning that the telomerase will tag, that is, they will add these things at TTA and GGG. Whenever you see TTA and GGG in your USMLE step 1 or your UWorld, try to understand that they're directing you to a telomerase question. The thing is, telomerase will go and they will add these things to the end of the 3' end of the chromosomes to avoid loss of genetic material. But if there's excessive telomerase activity, please write down over here. If there's excessive telomerase activity, then this can give rise to carcinoma, because excessive telomerase activity will result in excessive additions of these things, and the DNA replication will keep on happening time and time again, and no other sequence can go and stop the DNA replication. So this can result in unproliferated growth and differentiation, resulting in carcinoma. And there are some structures where telomerase activity is actually kind of low. For example, RBC, skeletal muscles, and those structures, but that's not high yield. But the fact is that telomerase activity, increased telomerase activity, is responsible for carcinoma, is extremely high yield. Okay, did you guys write it down? Yes, okay. Telomerase is basically TTAGGG. TTAGGG. That is telomerase. Telomerase is basically, what they will do is they will go and they will add these sequence at the end of 3'. When you add this sequence at the end of 3', then DNA synthesis begins and there is no loss of genetic materials. And this is a normal procedure. But if there's excessive working of telomerase, then there will be excessive DNA replication. If there's excessive DNA replication, there is a possibility of unproliferated DNA growth, and this can result in the formation of carcinoma. That's what I meant. Are we clear about this? Is this tailing? No. Okay, this is not tailing. Yes. Okay. Are we clear about telomerase activity and everything else? If we are clear, can we stop the lecture up to here for now and begin from tomorrow? Okay, so that we can do some questions. Okay, so that we can do some questions. It's so weird. When I ask you guys questions, I do not get enough reply. But when I ask you guys questions regarding can we stop the lectures, and everyone starts replying yes. Okay. And before, when I ask is this clear? Have you guys understood? Do we have questions? No one specified. Okay. All of a sudden, I ask you guys, can we stop the lecture? And then you guys, yes. Okay. 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 So telomerase is only working at 3' end and ligand between right now. Yes, only working at the 3' end. Okay. We are processing the concept. Oh, you're processing the concept. Okay, good. Okay, then my apologies. Okay. Okay. All right. So let's begin with some questions for today. Right. Before we start the questions, can we take a 5 minute break? Okay. Finally. Okay, is everyone back from their break? Is everyone back from their break? Okay, good. So how did you guys like today's lecture? Did you guys understand today's lecture? Did you guys enjoy today's lecture? I realize a lot of you get a little bit fatigued after 12:30, so I decided to start the lecture over here. But we have to finish the lecture. Can we start tomorrow very early at 9 a.m. exactly, please? Because we do not want to wait for 10-15 minutes for the full thing. Okay, so let's start tomorrow very early so that we can finish it on due time. Because what happens is there's so much information regarding genetics, and we don't if we don't understand this, and if it's after 12, I mean at least over here in New York, after 12, what I realize is that the response rate of the students, they go down very fast. And this makes me think that you guys are not really understanding the lecture properly, and you guys are just waiting for the videos, and then you guys will go through the videos to go through the lecture. But that's not the proper way to utilize a tutor. Okay, so I need you guys to utilize this opportunity as the best way as possible. So in order for us to do that, since we do get a little bit fatigued at 12, let's start the lecture exactly at 9 so that at least we get 3 to 3.5 hours of solid first aid reading. Okay. Okay. So with that being said, let's start today's discussion on UWorld notes. Okay, we will keep on adding the UWorld notes. Regarding the UWorld notes, how are we doing in terms of revisions of the UWorld notes? Are we just collecting them or are we revising them? What's going on with this? Because what I just realized is Bloom syndrome. I have discussed Bloom syndrome in UWorld notes. So surprisingly, none of us remembers Bloom syndrome discussion in the UWorld notes. Are we reading the world notes? Okay. Okay. Okay, let's begin with today. Okay. Okay. Once we begin discussing the UWorld notes, first and foremost, you guys are at liberty to unmute yourselves and provide me the answers. Okay. That's that. But in order for you guys to do that, please make sure that if you guys have any surrounding noises, please refrain from unmuting yourselves because this will hamper the concentrations, including myself and the rest of the students. So please refrain from doing that if you have background noises. And when we ask you the question, please make sure that when you give the answer, once again, there's no background noises because what that happens is, once again, it will hamper the concentration. So that's it. So, and if anyone is ready to provide with the answer, please say I'm ready and then begin so that we can give everyone a proper time to participate. With that being said, let's begin. The first thing that I want to discuss today is if we have, okay, if we have a defect in Sertoli cell function, okay, this results in the disruption of claudines and alkylates. Okay, as a result, what we have is when they get released, this results in the formation of anti-sperm antibodies. Okay, this was not a question, this was more or less a statement. Next one. Next one is, are we well acquainted with the word tachyphylexis?
Yes.
Yeah.
Repeated use of, first of all, what is tachyphylexis?
Sudden onset, not responding to the drug.
Yeah. Loss of function after repeated stimuli.
Meaning sensitization, basically.
Desensitization. Yeah.
Repeated use of phenylephrine can result in
Tachy.
tachyphylaxis. Next one. which procedure is performed to detect paroxysmal nocturnal hemoglobinuria? Anyone?
Flow cytometry.
Does anyone else have any better answer? Okay. Flow cytometry is the right answer. Very good. Okay. Tuberous sclerosis occurs due to mutation of
Hamartine and tuberin.
right. Very good. Hamartine and tuberine. But what are the chromosome numbers?
16 and
So we have TSC1 on chromosome number 9.
9. Yes.
Hamartine or tuberine.
Hamartine 16.
So what is this one?
This one tuberin. Yes.
Is everyone sure about this? Do we have any better answer? Is everyone sure about this? Okay.
First one is tuberin. Yes.
Tuberine. And TSC2 on chromosome number
16.
16 is
Hamartine.
okay. Next one. Next one is what is the okay? What is the leading cause of death in paroxysmal nocturnal hemoglobinuria?
Kidney failure.
No, I don't like this question. Okay, because this can go both ways. My apologies. Okay, calcification. Another question. Calcification of cholesterol crystals. Are found in the histology of craniopharyngioma. Okay, next one.
Mm.
Next. Okay. Do we have to prescribe vitamin D to neonates after birth?
Yes.
Okay. The reason is vitamin D in breast milk is not enough to prevent rickets. As a result, patients should receive 400 units of vitamin D per day. Do not forget 400 units. Okay. Okay.
Okay.
Mitral valve prolapse is seen on anorexia nervosa. Basically, patients of anorexia nervosa, they can have mitral valve prolapse, okay, because they have excessive loss of connective tissue. So that's that.
Mm.
They're not having enough protein, and if you have to have a cardiac dysfunction, the dysfunction is mitral valve prolapse. Okay. Next one. Okay, 1 second. Okay. In moderate doses, epinephrine increases beta and alpha agonism, and phenyl benzamine is a non-selective alpha blocker. This results in increased beta receptor agonism. So if we use epinephrine and phenylbenzamine, phenylbenzamine together, this will result in increased-
Tachycardia and hypotension.
right. So what this will do is this will have an increased
Heart rate.
Okay, the question was, how do you have increased synthesis of insulin in a patient who has used excessive epinephrine and phenylbenzamine together? So basically, the concept is very easy. One is an alpha blocker, another one is both an alpha and beta agonism. If you block the alpha, excessive beta working, the pancreas have the beta receptors. So when they go and act on the beta receptors, they have excessive insulin secretion. That's that. It's a very easy thing to understand, but I felt the need to add this to the notes so that you guys can make the proper connection. Okay. Okay. So on the peripheral blood film of Klinefelter syndrome. Peripheral blood film of Klinefelter syndrome. What do we see? We see
Barbadies. Barbadies
very good. Okay.
in neutrophil.
Dr. Noah, do you have a question? Abuse, would this lead to heart failure? Okay. Dr. Tasabi, where is this note? This note, I will provide this note for you. Have you not received this note from have you not received an email about this note? Yes or no? Have you received an email about this note, Dr. Tasabi? You can use the chat box to provide the answer. Have you received? Okay, if you have not received this, then I'll make sure that you receive this notes today. Okay. Peripheral blood film of Klinefelter syndrome. What we see is we see bar bodies. Bar bodies are nothing but activated X chromosomes. Okay. Okay. Why do we see bar bodies on Klinefelter syndrome? We see bar bodies on Klinefelter syndrome because in Klinefelter syndrome you have methylation of cytosine nucleotides. Okay, that's that. Okay, what are the drugs that can cause lithium toxicity? What are the drugs that can cause lithium toxicity?
Naside.
Thiazides
Oh.
and NSAIDs, except aspirin. Okay. 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 Yeah, okay, 1 second.
Doctor,
Yes.
ACE inhibitor can cause also lithium toxicity.
Which one?
ACE inhibitor.
ACE inhibitors can cause lithium toxicity. I'm not really sure about this. I'm not really sure about this. I don't want to give my. Let me just take one thing.
Yeah, they can cause.
Okay. Okay. Very good. I wasn't sure about this. I don't know why. So I guess ACE inhibitors are also there. Okay. Thank you so much. Okay. Very good. Okay. Okay. Okay, the drug that is ticlopidine. Ticlopidine is it's a P2Y12 receptor antagonist, which will result in decreased platelet aggregation. Okay. And Okay, that's that.
It's same like clopidogrel, yes?
Right, correct. Clopidogrel and ticlopidine, same drugs. Okay, what is the first line drug for hairy cell leukemia? Anyone? We talked about this before. Aren't you glad
Cladribine.
Cladribine and
everyone can
pentos,
say everyone can
pento.
say cladribine after I have wrote cladri? Okay, so cladribine, very good. Nonetheless, thank you so much for giving the right answer. Next question is what is the mechanism? Okay, this is not the mechanism. What this is cladribine. They are resistant. Okay, 1 second, please. Okay, my apologies. I'm sorry. Okay, cladribine, once again. First line drug for hairy cell leukemia. Cladribine is resistant to breakdown by adenosine
Yeah,
deaminase.
mayonnaise.
Did you guys know about this?
Yes.
I think I got
Yeah.
this question. Yes.
Okay, if
Yes.
you guys knew about this, what is another drug that is resistant to adenosine deaminase? Adenosine deaminase deficiency. Although this is a drug which is high yield for step 2 CK, but not for step 1. Adenosine deaminase deficiency, sorry, adenosine deaminase resistance.
Pentostatin.
Pentostatin. Did you say pentostatin?
Yeah.
Oh, very good. Okay,
Both are used against hairy cell leukemia.
right. Cladribine and pentostatin. Okay, very good. Okay, we're very high yield for 2 CK, but not for step 1. But sometimes you may receive these questions to differentiate between extremely good marks and bad. I mean, the regular marks. So that's that.
Hmm.
Okay. Okay, next one. Okay. Chloramphenicol is high yield for causing aplastic anemia out of all the drugs. Next one. Um, 1 second. Okay. Circular. I'm pretty sure you guys know this. Circular's bullseye lesion in a patient after camping. What is the bacteria?
Lyme disease. Borrelia burgdorferi.
Okay.
Oriella.
Okay. Who are the vectors?
Tick.
So this.
exodus.
Okay. And the reservoir. Who has these tick?
Deer.
Deer. Okay, upon my apologies, I cannot hear someone trying to say, can you guys use the chat box? One student said deer. Do we have any other answer?
Yes, cattles and
Okay. Next one is in chest examination, if you hear scratch and high pitched breath sound. Okay. Scratch and high pitched breath sound. This is an indication for pleuritis. Okay. Okay. Next one. Increased MAC, meaning minimum alveolar concentration, means increased potency of a drug. Increase. Okay, that's
doctor
that.
mean increase potency or decrease?
Increased, right? Minimum alveolar concentration, meaning the less
Decreased.
amount of drug, is needed to cause 50% of
Okay.
to undergo.
less.
Okay.
Yeah.
Okay. Sometimes when you guys ask me questions out of the ordinary, I have this habit of being very. I don't know if you guys have realized it, but I have this habit of being extremely involved in a single thought process, and when I get and then when I get asked another question, it takes me some time to realize. Okay, so whenever something like this happens, I apologize if it takes me some time to answer this, but hopefully I will always provide the right answer. If not, please let me know if my answers are not right. Okay. Next one is Heberden's and Osler's nodes. Are they tender or not tender?
Tender.
Tender.
Keep that in mind. Okay, Riedl's thyroiditis can present with lower limb pulsations. Lower limb with I'm sorry, with increased pulsations of lower limbs. Okay. Okay.
This is the chronic, yeah?
Yes. Most common site of MRSA colonization. It's in the anterior nares. The next one is corticosteroids. They will decrease NFKB and phospholipase A.
Phospholipase A2.
Okay, I'm going a bit fast because I want to finish this.
You put phospholipase A strudel.
Right. I'm sorry. Okay. Okay, this one is my last one for today. Okay. My apologies for adding so many questions. Last one. Have you guys ever heard of a ganglion cyst?
Yes.
Yeah.
If you guys do a histological examination of a ganglion cyst, what type of cells are you supposed to find? I know find synovial cells and collagen and this and that, but my questions is it's a type of connective tissue, right? This is the synovial.
Yes.
Is it a monocellular or paucicellular? What do you mean by monocellular and paucicellular?
Paucicellular.
Right. Okay. And then there's another one. Okay. Why do we find cellular? What do we mean by that? What do we
Also
mean
mean
by
multi.
Yes, of course. But what happens is that the ganglion cells, they contain a multiple of cells from different types of synovial fluids and amino acids and cartilage and this and that. So the answer is, who was the first student who said paucicellular?
It was me,
Okay. Who is me?
Dr. Tureen.
Okay, thank you so much. Very good answer. Multicellular connective. Okay, perfect. So that's what it is. So we have
Doctor,
type
I have a question here. How
Yes.
could radial thyroiditis cause lower extremity pulsations, increased
renal
pulsation?
thyroiditis, they can present with increased pulsations of the lower limb. Uh, the thing is, for sometimes the thyroid, they can go and redox thyroiditis. They can increase the, so what happens is redox thyroiditis is fibrosing thyroiditis. So it
Yeah,
increases the pressure. I mean, it increases the resistance on the blood vessels on the upper part of the arch. So it's easier for the blood to flow to the lower part of the body. So instead of going to the upper part as much in redox thyroiditis, it can go to the lower
it is
limb.
diverting the flow.
The flow can get diverted. Okay, this is very this is not high yield. Okay, always remember that most of the ones that are over here, some of them are not high yield, so some of them are just there because it's an anomaly question, meaning that it's an abnormal question. I mean, it's not normally, you will never see a patient with increased pulsations of lower limbs in a thyroid problem in a clinical setting in a hospital. Okay, but if you have to do extremely well in your exams, sometimes you have to take these into consideration. So that's that.
Yeah,
That's why
Yes.
I.
thank
Okay.
you.
Doctor,
Okay.
can you mention the ganglion cyst in last note?
Paucicellular connective tissue.
No,
Ganglion
no,
cyst.
it's not written, just histological examination. So in future when we
Oh,
read,
histological
Yes.
examination.
Yes.
Ganglion cyst.
and one more question, where we can find this cyst, like what is the origin?
Oh,
Because-
ganglion cysts. The ganglion cysts are basically, they're found along a tendon.
Tendon.
They get formed in the hands and the feet. Okay, let me show you a picture of ganglion cyst. I'm pretty sure a lot of students, I mean, a lot of some students have seen the surgical setting along the hand area. So what happens is ganglion cysts are basically, they're basically collections of synovial fluid. And it's a very benign condition, non-painful, non-tender, and they can be surgically removed or they can be follow-up with a procedure, and they have a tendency of being resolving on its own. You can either do a cystectomy or you can do an aspiration, either one. Okay, are we clear?
Thank you. Yeah, thank you.
Okay. Okay. If all is well, can we end the lecture here today and do the questions tomorrow?
Yes.
Yes, please.
Okay, once again, everyone say yes, yes, please. Okay.
Can you send the updated document?
Okay,
Okay.
perfect. I'll send you guys the updated documents. Thank you so much for doing the lecture for today. I want to end the lecture a bit earlier today, but tomorrow I want to start it a bit before, but it's already 1, so it should not be that much of an issue. But since we could not do many questions today, but we added a lot of UL notes, we'll try to do more questions tomorrow, unless UL notes. We'll try to do around 5 to 10 questions tomorrow. Is that okay? Is that okay, guys? Okay. All right. So with all that being said, so much questions from biochemistry, from the genetic portions. Okay, hope you guys have a great day today. If you guys have any questions, let me know. I'll answer them for you. And I'll upload the UWorld notes and I'll send them via email. All right, thank you so much. Hope you guys have a great day. And I'll see you guys tomorrow at 9 a.m. exactly. Okay, please do not miss out on the biochemistry lectures. Please do not wait for the video because it's important you guys be present in the lectures and ask me questions so that your concepts can be cleared. Okay, please make sure you guys use the tutor that is us in the best way. Okay, and that's by being present in the lecture. All right, with that being said, thank you and healthy life. Bye-bye.